Ravi? Good evening, everyone. A warm welcome to all doctors who have joined us in this evening program today. First of all, big thank you to all of you for taking time out on a Friday evening and joining us for this G.R.O.W Scientific Symposium. So this is the second program. So we had one fantastic program yesterday evening in Delhi. So with that, I would quickly give an overview on the aesthetic business of GUFIC. So coming to the purpose of our aesthetic division, the purpose is very simple: to make a difference to our customers, to our aesthetic practitioners, and of course to your patients and your clients. So as far as vision and mission that we have looked into for our aesthetic business, the vision is to be a trusted leader in dermatology space with excellence in innovation and of course driving very strong innovation and excellence with a focus on aesthetic dermatology. And our mission is to bring you the best in terms of differentiated and innovative products which is going to come in the future. And we are committed as a team to build a very quality-driven team in place so that we can collaborate with customers and clients to enhance their knowledge, skills, and their practice. So coming to a quick overview on what's our strategy and the portfolio that we would like to build as far as GUFIC's offering is concerned. We already have a botulinum toxin in place. We are looking at building our portfolio with a world-class filler, which is going to be launched soon, which is a good news, I would say. It's a US FDA brand that we would be launching in probably 6 months' time from now. And to supplement that, we will also have a skin booster range and biostimulators going forward. This is just going to be the start, and subsequently we are also looking at meso-based complementary products, because as a company, GUFIC has specialized over the last 50 years into lyophilized injectables, and that's our forte, and we would like to continue to strengthen and build on this important segment that we are in. We already have set a new standard of innovation with Stanox, which is the first and the only Indian brand to be made in India, which is in technical collaboration with Prime Bio. We have Dr. Balram sir today, and it has been possible because of the collaboration that we have had with sir and the senior management at GUFIC. And of course, it has a lot of interesting aspects to it, which I would definitely leave to sir to explain us the details. Stanox has become a new standard for stunning skin in India already. In the last 4 years since we have launched this brand, since 2021, we have had an impressive journey of milestones, right from the time we got the US government permission to import the culture and the import permission that we received from the Indian government, right to the storage of this very important culture, then harvesting it, and then lyophilize the product that we have finally launched in the market. So just a very quick snapshot which I've shown here, but more in detail, I would request our CEO, Mr. Pranav, to talk about it at length. The first and foremost milestone on Stanox is that more than 50,000 patients have already experienced the benefit of this brand, and we are going from strength to strength. What is very important for all aesthetic practitioners is how the market is poised to grow. One good news is that for the next 5 to 6 years, as per various reports that are available on aesthetic market, this category, especially on the toxins and the fillers, is going to grow by higher double digits, and which is what you can see in this chart. Both toxins and fillers are going to see that rise continuously for the next 5 to 6 years, which means that in the next 5 to 6 years, the market, which is currently around 35 million, which is around 300 crores approximately, will be double. And that's a big opportunity for all the practitioners out here. And GUFIC as a company is putting all the best foot forward in making, or rather partnering with the doctors to make it happen. And in this market, which is the botulinum toxin market, the good news is Stanox is already the +2 brand or the most recommended brand in the country. Now, what is actually making this possible? We have a 40+ member team all India, which is led by a team which is represented by Madhu, Ravi, and his team, the medical team by Dr. Jyoti and Dr. Rajesh, whom you would have all interacted in the past. So I would like to now welcome Dr. Jyoti to talk to you all about the G.R.O.W initiative that we have already introduced to the doctors. And I now invite Dr. Jyoti to take the proceedings forward.
So a warm welcome to all of you. So when we talk about G.R.O.W as a medical initiative, we are telling you that we are here to grow your skill set, grow your expertise, to basically empower you as an injector and to create an impact. So when we talk about our mission is to empower dermatologists and plastic surgeons through scientific excellence and hands-on training in injectables aesthetics, fostering a community of safe, skilled, and confident aesthetic injectors. Our vision is to be a leading force in aesthetic education, ultimately redefining consumer trust and expectations in non-surgical facial rejuvenation. When I think of G.R.O.W, the core objectives for us is scientific training and skill development, which is a need of the hour for all us as practitioners. Standardization of aesthetic practice for ensuring consistent and high quality outcomes across all the clinics. Building a safe injector ecosystem to create an impact in Indian scenario for our patient satisfaction. Innovations and research integration. GUFIC is known for in this particular aspect of research and innovation, thus making sure that we generate more of clinical data which is focusing on Indian patients. And patient-centered care, where we take care holistically at the level of consumer, create education and awareness. See, because if there is no education awareness at the level of consumers, so how do we grow as a practitioner? So G.R.O.W is going to be focusing on all these aspects, and as per the survey, it was proven that today if we want to be enhanced and upskilled, the most important thing is robust training programs. Qualified trainers are the most important requirements for HCPs in India. So on basis on this, our G.R.O.W initiatives are going to be focused on strengthening the medical education and training for each one. So when we understand Indian scenario, our advanced practitioners under the banner of G.R.O.W, we want to make sure that we build, engage and educate through robust training one-to-one interaction. We make sure that we develop a strong conviction and engage them as advocates for the science. Not only, it's not limited to a product. And generate Indian patient data through clinical studies and publication. Trust me, I think there are very few doctors who have really worked into this realm. Today, almost more than 2.5 decades, especially when it comes to botulinum toxin, we are all dependent on, the companies which are, of course, not Indian. And the database, the patient studies is nothing which is our ethnicity. So how do we, make a strong base out of this and give opportunities to our doctors to help us develop this clinical data and publications? And exposure to global educational initiatives, we feel we have such an immense talent pool in India. And we want to create and foster that community of doctors to give them that platform on international congresses or internationally where they can update Indian scenario and the Indian results and outcomes. And also, if we have to grow, we also have to make sure how do we support our young budding dermatologists into training them as a leading partner to train them and promote them as a safe injectors. So we are under the Grow, we want to have this Grow Them Young program where we want to collaborate with national associations and bodies and develop a very strong focused module based program for them, where we focus on assessment of the patients, how do they understand the anatomy, the injection techniques, and also to manage complications. So one of the initiative this year we have started is in-depth clinical understanding and symposium based on botulinum toxin. I'm sure we all are very aware that when we talk about botulinum toxin, somewhere as practitioners, in the whole race of patients and clinic and practice, sometimes we miss the thread of science. And it's very important that we go and we dive sometimes very deep into it to understand what are the possibilities with botulinum toxin. And with this, we are very proud that we have invited Dr. Balram Singh, whose 37 years of experience and work is like a great, I think I have very, very blessed to be, learning in last few days with you, sir. And I think it's an immense opportunity for all of us. Along with him, we also have Dr. Andrew Ress, who has a lot of cadre of work into botulinum toxin along with Dr. Singh. And we have such amazing panels today, panelists for y'all, to discuss the Indian experiences. How we Indian patients are very different. Is everything same when it comes to the different ethnicity? So yeah, we will have some interesting discussions. So I welcome all our panelists also. This is just a quick overview of our agenda today. And with this, before we begin our program, it all starts with a vision and a right leadership. With this, I would really want to invite Mr. Pranav Choksi. He is one of the most dynamic leaders I have come across, and very fortunate to learn from his experiences and his passion. Mr. Pranav is a Master of Science in Biotechnology from Johns Hopkins, continuing the 50+ years of family legacy of pharmaceutical innovation, and that introduced, I think, many antibiotics, especially oxytetracycline and amoxicillin, to India. Revolutionized healthcare with innovations in dual chamber bags, novel HMG purification techniques, scaled manufacturing capacity from 50,000 to 1 crore vial monthly in under 10 years while maintaining rigorous quality. So if I have to understand in a very simple word, GUFIC Biosciences is currently the largest producer of lyophilized injection in Asia. So I think it's a very, very proud thing when it comes to, especially in the domain of critical care, infertility, and pain management. I think GUFIC is amongst the top players in the country. So a hearty congratulations. And driven by the vision of making advanced healthcare products affordable and accessible to all patients. So yeah, with this, I think I would want to now welcome Mr. Pranav Choksi to share his vision and how such a molecule like botulinum toxin to come and manufacture in India. So yeah, the stage is yours, Mr. Pranav.
Thank you for the exceeding praise, which I don't deserve. I think, like, as a sponsor, it's mandatory to have me, thoda acha bolna padta hai mere, so thank you very much. No, but it's very interesting. Thank you, I think. Thank you, Dr. Jha. Thank you, Vijay Bhai. Today, my interaction is to actually, first of all, in a nutshell, convince you that the product which we offer is really, we have really spent a lot of time, energy, resources, and a lot of scientific, I would say, brainstorming behind it. Today, in the field of aesthetics, when you come and, what do you expect? You expect the best for your patients. You expect the best techniques, the best product. And you also, the patient has very low chance of failure at you. Otherwise, it's like make or break. Standing against allergens and the Galdermas and others of the world, my only purpose today would to tell you, and the presentation would be in two parts. One is what is GUFIC, so you know that you're dealing with a company which you can trust. And more importantly, my journey, how I got Stanox or Zarbot. Stanox is a brand name of a product for aesthetics in India, and the same molecule is known as Zarbot for neurological purposes in India. And how did the journey happen? So I always tell people I have 3 kids. My daughter is Meera, 14 years old. Second is Stanox and Zarbot, which is around 4, 5 years old, and my son who's around 4, 5 years also. So I'll just come to that, a little bit of, I think the slide show, thank you, So again, I'll not spend much time on the company. I'll spend more time on the Stenox thing. But just to, like she said, my journey with Stenox started for a reason. I did my master's in Johns Hopkins. My specialization was autologous cancer vaccines. At that time, in 2002, Botox was approved by the FDA for aesthetic purposes. So I have seen the euphoria of Botox happening in US during those 4 or 5 years I was there. And it was something which, had, kept some impact on me, which I thought if I come back to India, I should work on it. So holding that thought there, let's come to GUFIC first, and then I'll come back to the Stenox story. My grandfather started this company way back in 1970s. Why is it relevant, I'll tell you. In 1998, the company was ranked the 22nd best company, by revenue, and known for its products. I don't know if you must heard about Mox and Zol. These are old products. And then there was Stretchnil, Roll-On, and all these other products which we had. In 98, my family decided to sell the entire pharma business to a company formerly, now known as something else, but Ranbaxy was a very big company. A lot of respect for that company, one of the truly first multinationals of India. We sold the entire pharma company to pharma business to Ranbaxy, and Ranbaxy to Daiichi and Sun Pharma. So from 1998 to 2008, we had a non-compete. So I could not do it. We, as a family, could not do anything. In 2008, I came back from US, and the first thing we did, we started the company from scratch from a 4 crore revenue, which was a legacy of Sallaki, which we had as a brand. And now from 4 crores to a 810 crore journey in the last, I would say, 16, 17 years. It took us a long time to touch our first 500 crores, now 800. And I hope in the next 3 to 5 years, we'll be touching 2,000 crores because of the reasons I'll explain to you why not. And going forward. So again, as you should be happy to know that we are a company completely backward integrated from API, formulation. Our major 55% of our revenue comes from India, but majority of our revenue comes from UK, Germany, Canada, South Africa, and Brazil. So that is where we are trying to become more and more global and bring those global standards in India. So a patient in India, a patient in Germany, or a patient in even Canada gets the same product from the same batch and same, I would say, production scale. These are just the different divisions. The history of GUFIC is such that we first came in critical care. So when I came back from US, I realized that pharma business is gradually becoming a commodity business. I think it's a fact because things where the entry barrier was many years ago, it could easily be replicated because all the governments are promoting generic drugs and they're promoting, ease of use and better pricing and all that. So there was always a thing inside me, get into those factors where the entry barrier is limited. So why critical care? I knew that there would be certain new injections, patents, drug delivery systems like dual chamber bags, which we as a company should work on, which makes us different. Same thing with infertility. So just to share, we are now the #1 company in critical care in our represented market in India. That's our first primary division. In fertility, we are now the #4 division in the #4th division in India after the multinational in India. But hopefully, we should be #1 in the next 5 years because of the manufacturing capability. And the next would be ortho-gynec. I'll focus more on the aesthetic part. The aesthetic business is something which I felt that even after so many years, there are only 5 to 6 global players in the botulinum toxin market. And what we see as botulinum toxin in aesthetics, there's much more application beyond that. And I and Dr. Singh both will try to, I would say, share more thoughts on in the next few slides. This is a very important slide. You should know, and it's something very close to my heart. So when I came back in 2008, I did not have much money. My dad had a lot of money, but he refused to bet on me at that time. So I started my journey as a contract manufacturer. So, I used to have my bag, and I used to go around from company like Abbott to Zydus and all those companies mentioned here, कि चलो मैं vial injection बना के देता हूँ. And that's why it is very important because this was my growth story in terms of regulatory, in terms of pipeline, in terms of quality, which I learned in the first 10 years of my life, which I can now reciprocate. So that is where that 50,000 vials to 1 crore journeys happen. For a company of, so just to put that into relevance, 1 crore patients are being served on a monthly basis by us, by GUFIC itself, just the injection market. And this is not only in India, but in the world. And that is something which is, and this scale is really crazy because the next company after us is a Chinese company, which is almost 70% of our capacity. Only thing is that we need to move more, make the pipeline more relevant in terms of new additions, which I'll talk about it. Again, I'll not bore you with all the, I want to spend more time on Stenox today, and the journey of Stenox. Just like I said, we are present in more than 40 countries. Europe, and there are two things which you don't see right now. That is the US and the Japanese market. But that is because we are going to enter very soon under his guidance and from our Indore facility. Those two Gs will come in US and Japan also in the next 2 years also. Again, I'll not bore you with that. The infrastructure. So the infrastructure, what we have, and that is where is going to be one of our topics going forward. Don't believe whatever I tell you today. Today, everyone comes and, this is a platform I'm expressing myself, but you should come and see the world-class infrastructure which we have at Navsari, where currently your Stenox is being made. And now the new infrastructure which is coming up at Indore, which is going to take it to the next level for the US markets also. So these are real cutting-edge, I would say, automated, non-manual intervention factories where there's no human intervention. The issue is a botulinum toxin is a live toxin. How we get cough, cold and all that, even toxin is prone to bacterial, fungal and also other infections. And that is how this technology and this capability is very unique as GUFIC, that we can ensure that we get you the right product in the right way. Again, I'll talk about it in the next few slides. Okay, and I'll not bore you with all this. I'll come straight to, the last slide is important. So the secret of GUFIC, why a big company? When I say a big company, when we started the vision of getting botulinum toxin to India, our revenue was hardly around 250 crores. So it was in 2015 to 2016. Then why a big company, and there are top 10, top 20 companies, why did they never try to get a botulinum toxin into India? Was it that they didn't have the money? They had more money than us. What is that? They didn't have that need for that product. They had it. The only thing which was missing was identifying the right talent and also having the right skill set in terms of biotech. And that is where we are quite proud, and not me, this is not my skill set, it's my team's skill set and also skill set of Dr. Singh, who's one of our important part of our team, that we always try to do things which are differentiated and unique. So when nurses in India were mixing a vial by doing a diluent and a vial, like you also do sometimes, you have an ampule, like we have a botulinum toxin, you have a film form still, you have a plastic thing or you have a glass ampule and then you have glass vial, you break it, you mix it. All this is very far. So as a first time in India, we came up with the dual chamber technology, where you have an entire chamber, just mix it, and it's like a 2-chamber. You push the water into the powder, mix it, and you give it. I think we'll have samples for you next time in the field. We'll show it to you. One more technology we worked on is dual chamber syringe technology. So dual chamber syringe, where you have 2 chambers in the form of a pre-filled syringe, where you have a powder in the front and you have liquid behind. You just push it ahead, the liquid mixes the powder and goes in. And this is the technology we are trying to do for Stenox going forward. So in Stenox, the problem of reconstitution, the problem of mixing, not getting the right dose, sometimes instead of 2.2 ml, 2.5 ml, you do 3 ml, why? Everything will be much more thing. If you want tomorrow, a doctor asks me, I want 10 units ka PFS with me, so I know I make no errors. Or I want only 1 unit ka PFS titrated, so I just mix it and just do it. So we are working on a technology of thin film PFSs, where the thin film is there on the front part of the syringe, the liquid is behind, and you give it a unique shot of 1 unit, 5 units, 10 units, and so on. So this is something, everything comes with the IP protection, because I feel in business, if you have to grow, you need to have a IP created, value created, then only it makes more relevant sense. And just as a slide, so all the way you see from EU, Canada, like I mentioned, except for US and Japan, you see all the accreditations here. So the product coming to you is coming from a facility which all these countries have come and inspected and have approved also. So there's one more point of confidence. Now my favorite part. This was the boring part of the presentation, my favorite part of the presentation. Are we ready with the next slides, please? So now I'm going to talk about my journey of Stenox. So when I came back in 2008, 2009, I wanted to get desperately botulinum toxin into India. Desperately. Just because at that time, very frankly, it was a fad. Let's get botulinum toxin. I want to get Botox. So the first thing I went to Allergan when I was there. I'm shifting to India. My family has a business for so many years. We want you to license this product through us. And I believe they were already there in India at that time, very frankly, when I was in 2000. I didn't know about it. I was a novice. I didn't have any knowledge. And they didn't even answer my email. It was, I mentioned, in the trash can somewhere. Then I went to China. Normally, as Indians, the first thing for sourcing, we go to China. Kuch nahi mil raha. So I went to China first. From China, then I went to Korea. I went to Israel. I went to Italy. So from 2008, my search kept on going on till 2014-15. And I almost had given up. And then in 2014, I met my guru, my mentor, another friend of mine. I'd call him also my guru, Dr. Verma. He's aware about it. I know him from the US days, and he was one of the important professors in University of Connecticut, if I'm not mistaken, who was working on liposomal amphotericin and liposomal technology. And we were working with him for liposomal technology. In one of the evenings, I said, sir, I'm desperate to get botulinum toxin in India. He said, oh, you should have told me before. I know Dr. Singh. He's my friend in US, and it's serendipity. He said that, why don't you contact him? So it was 31st of December. I really partied hard. On 31st December 2014, I partied till 2:30 in the night. In morning 7:38, I went to his hotel to meet him. I think it was somewhere in Bombay. And I said, sir, I'm desperate to get botulinum toxin in India. And then we kicked it off. I mean, I really had to woo him to convince him that I'm the right partner for him. He was a gentleman at that time, working on botulinum toxin for almost 29, 30 years, working on the strain, the pure science, and I was blown away because I think it's like I had hit the jackpot, because I thought I would just get a product made somewhere, get it here, then do a tech transfer and just make the type A. But here I had the mothership, where you have a person working on the strain of botulinum toxin for last 30 years, and he's not only ready to work with me for the type A, which you all use right now as Stenox, but we are working on solutions of liquid formulations and 6 to 8 months longevity and type E and topical formula. So this entire pipeline is something which we discussed on the first day itself. So that's how I went and met him in US in 2016. 2016, we signed an agreement. 2017 is where this journey starts now. Like I said, my second kid, because my first two kids, my first and third kid took 9 months. This kid took almost 7 years to come, I think, in the market of India. So as Vijay Bhai said, the journey starts because, as you know, 1 teaspoon of botulinum toxin, actually the extract can kill the world. So that's how potent and toxic it is. So to handle that and handle it in a decent amount of quantity is really complex. So first we had to go to the US government, and luckily he used to work a lot with the US Army. He still works a lot with the US Army because it is also used as a bioweapon. It can be used for terrorism and other factors. So first we had to convince the US Army that we are exchanging this strain to India, give us the permission. That's why you have the Homeland Security also as a part of it. And we got the strain in 2017, I believe. That's how it started. And from 2017, managing the US, then we manage India also. We have to convince the Indian government that we are going to do some good things out of this. So we got the permission from them to import it. And mostly I have kept the presentation as photos, so it's more fun. Otherwise, it becomes just text. So especially for my $1 billion baby of botulinum toxin, we had to get this entire container, which was at, I would say, 228. Inside that was this single box, as you see on that side. I think, this single box. So that's the only thing which came all the way from the shores of Massachusetts to Mumbai, Maharashtra, and then eventually to Navsari, Gujarat, near Surat, where this was unboxed. So in this entire box, in that entire container, this box was there. And in this entire box was this relevant box. And in this small box was this, which was my future $1 billion idea. So that is how the journey started. This is known as the master cell line. So the master cell line is the most important part of it, because if you see, a process goes. Botox or any other brand was done either in the late 1990s or in the early 2000s. The genetic sequencing of that strain is very relevant. Luckily, when we started working in 2015, we already knew the strengths and the weaknesses of those strains. So when I mean the strengths and weaknesses, I'll only focus on the clinical part of it. The onset of action. How long does it last? How potent is it? What is the dispersion? What is the immune response? What sort of an inflammatory response is produced in the body? And then after multiple use, what will be the effect? So all those data which is there with him in US helped us select the right strain. So if you see 96% of our strain, genetic strain, when I mean strain means AUGC, that's the adenine glutamine, the entire genetic code is same as Botox, but the 4% which we changed and we selected, that is the thing which is going to make that difference. And again, I think more than me, he'll do more justice to that description when he comes or in the Q&A round. And that is how we selected the right strain. Now, just selecting the right strain was not enough. The master cell line is kept in a facility in Navsari in Gujarat, which is having motion sensors and so much security measures, because it is guarded with our lives. Because again, I'm saying, apart from the terrorism part, if anything happens to this, the entire thing goes for things. So the master cell line, say we make a working cell line, and the master cell line is kept outside. The working cell line has to be tested also for bacterial, fungal, and viral infections. Like I said before, all is a live strain. You have to keep on checking it regularly. It doesn't mean you have checked them in year 0. That means in year 2, year 3, year 5, it's going to stay the same. Like all of us, we change over with time. Unfortunately, there are mutations in our body. There are also mutations in the strain itself also. So each and every working strain, the working cell line or the master cell line are checked on periodic times for all these infections and so on and so forth. So from that strain, we made a working cell line. From the working cell line, we made a concentrate. From the concentrate is where the actual, I would say, tattva or the main ingredient came, which makes your actual injection. Now, to ensure that the product coming to you is the right product, we have to do a lot of tests. So like I said, one is the microbial purity. The second is the genetic purity. The third are the phenotypic parameters also. You have to ensure that there is nothing wrong with the strain. So each and every strain of yours went through a wrong process till it becomes an injection. This is Paul. Paul was part of Dr. Balram's team who also worked on this initial project. And he's still working on the project with the new products. And that's how the entire thing, I'll just a little bit skip the slides. This is more boring, but I'll come to the interesting part to show that 150 kilodalton and the 750 kilodalton is relevant. Also, there'll be some questions in your mind. A lot of people talk about 900 kilodalton as a complex. Some people talk about the pure toxin. What is the difference? Why, what is better? I'm sure everything has their own strengths and weaknesses, but we have to be clear. We have to today explain to you scientifically what is the rationale behind the complex and not the pure toxin. And that is why. So in our product, of course, we have the complex, 150 plus 750, and that we had to identify from this equipment. That is the ACTA from GE. And then even after identifying the genetic purity or the protein weight, we also had to done a lot of Western blot, or we had to do some gel electrophoresis to show the molecular weight is still relevant. So getting the 150 kilodalton was very crucial. And then, of course, you have other things which you have to remove. So that further goes to several steps of purity to get into the robust product. More importantly, you also have a question in your mind, if I keep on using botulinum toxin, does that have any immune response? And is that immune response going to differ in different genetics, or is going to differ in different patients also? So the product also goes through a lot of different tests of immunoresponses also. That's why you have the standard, and you have the sample. So this test, all the products coming to you, the cell line, the concentrate is also checked for several immune responses, keeping in mind the Indian DNA. Also. And of course, when I mean Indian DNA, it's an add-on. We also take care of the Caucasian one, the African, and so on and so forth, because this is a product designed for a global platform. Again, Western blotting, protein determination, these are all different tests. And then the most important test, which looks very scary, but it's mandatory. The IU which you see in, in your sample pack, that is a 50 IU or 100 IU, that depends on the LD50. That's the lethal dose 50. So you have maybe around 10 subjects or 20 subjects. The minimum dose required to kill 50% of them. And that is where we know that it is potent enough to have the desired clinical effects. So this is the last step. This is a more conventional step, and we have also now better steps to go forward, because I feel in the next 5 years, the PETA or the cruelty to animals will force us to get into things where we have to go beyond animals also. But as of now, better them than humans. Down the line, there might be cell lines which we will come up with, which will make it more easier to avoid, to spare these innocent creatures. So these are just the photographs of the different machines which we use in the entire process. Just to show all this, of course, today I cannot take you to Navsari, but I would like to bring Navsari to you. Hopefully down the line you should go and visit us and to actually check that where your products is being made. Because when you have to change from a multinational product to Stanox, you should also be aware it's a multinational and the same global quality product also. These are just the different products. I'll just come to the next point which is relevant. Now, what we have always been known for our manufacturing of injection, which is our USP, again we have to say. So today, even during the, when is the toxin the most, I would say, vulnerable, is when we dilute it in water. And that is time, it is where this automated technology and non-human intervention makes sense. So all these equipments which you see looks complicated, but this ensures that the toxin once made, till it reaches to your clinic or to your enterprise, there is no human touch at all. Everything is happening via machines. One side, the vials are moving. This is where the actual toxin is being put into water, sterilized, made it into a solution. The other side, the vials are being washed, sterilized. And more importantly, they both come together in a liquid form. Before it becomes a thin film we talk about, it is actually a liquid, which is form of a vial, which is open. And then, as we all have ovens and pizza ovens at home, this is actually a lyophilizer, where one shelf of 1,500 vials goes in a shelf, and then we put it inside a lyophilizer, or like, let's call it a freezing oven. And then there's a special technique by which we want to ensure that we want to keep the toxin alive, but at the same time remove all the water out of it. And when we do that, it's known as the process of lyophilization, where we take it to the freezing point. But anyway, that's boring. But the more important point is, you have the most potent form of toxin alive in your vial for a period of 36 months to 48 months. And sometimes in the future, you will tell me, "Abhi 2 to 8 bahut ho gaya. Abhi ek baar below 25 या below 30 degree का भी product दे दो." That is what we are working here. So even tomorrow, if there's no, I mean, you all are from Mumbai, so not to worry. But when we take it to the interiors of India, there might be no electricity. There might be no control. There might be no access. There might be products which might be reaching after 5 days. How do we take care during that logistics? That is what the studies are all done based on this lyophilization technology, which makes it more pure, potent, and stable. And one more thing. Everyone has heard about sodium chloride. A lot of our competitors use sodium chloride where the light chain and the heavy chain breaks, which I think Dr. Russ will also talk about. Ours is a very unique product, and why we say a thin film technology, we have the human serum albumin, we have the citric acid and the sucrose, which is very unique, by which even if a product kept for a period of, right now I think it's 36 months, but very soon in the end of the year, you will start getting a 48-month shelf life product also. Ensuring from day 0 to day 48 into 12 into 365, whatever it is, 48 into 30, sorry. The day the potency is the same. And that is what we are working on. That's again with the help of Dr. Singh and our team. So this is all possible because of this thin film technology, what we have. And again, this is the capping. And that is where, voila, you get the final product. More importantly, right now, the standards which we minimum adhere is the British Pharmacopoeia and the European Pharmacopoeia. But we would like to say that we go and we perform tests beyond what the government and the industry ask us. So with Dr. Singh, we are luckily exposed to so many testing mechanisms, like the immunoresponse is not mentioned here also. Even the other tests of bacterial and fungal purity is not mentioned here also. But we still do it nevertheless on every batch, on everything coming to your clinic. Even the vacuum test, we have a lighthouse instruments, which is, just to explain to you, if you see a vial, you have a thin film below, and you have the head space. We call it a head space. A lot of people fear a vacuum nahi hai, what will happen? So each and every vial of yours is tested in a lighthouse instrument, where because of the oxygen setup or because of the vacuum, if the current goes through, that means there is nothing. I mean, if the current goes through, there's a medium for the current to pass through. When the current is stopped, then you know that there is something like a vacuum where the current cannot go through. So each and every vial is tested for that. And now we are working on a product that, be it vacuum or be it temperature, the product is going to be stable. I'll again leave it to you when you come and present that. So all these tests are actually done on the product before it comes to you. And again, why I'm saying this is just a start. Right now, you have a lyophilized injection which helps you for 4 or 5 months. We are working on products which will be a topical form. Again, a topical form doesn't mean that the consumer will be able to take it on its own. We will still need you as experts to apply the topical form on the patient, because if it goes into the mouth or if it goes into the eye or somewhere, it still can be very dangerous. But the only thing is the topical might last for 1 month or 1.5 months, depending on the studies which we get in the next 1 year. Then we'll come with a type E, where there are certain sports injuries where you want the product to be paralyzed, or you want to go to any event in the evening. We give you a type E shot in the afternoon. In 1 to 2 hours, I believe, the effect comes like a normal type A toxin, but it lasts only for 2 to 3 weeks. So these are different applications we are working on as an extension. The most interesting part, we are going to come back to you, is in the next, I think, 2 to 3 years, because it requires a lot of phase 3, is a product which will last for at least 8 months. So this product is going to be a game changer, because the important thing is not to give you a product. Right now also I can give you a product for 8 months. But the problem is I should give you a product which lasts for 8 months, at the same time doesn't have any side effects in the patient's body. Because for us, it's not about giving you the potency, it's also about ensuring it's a safe product, but there's no other body mechanism getting altered by which the patient gets any sort of side effects on the long run or any mutations also. So those are all the works which are happening in the background at GUFIC, where really we want to tell you that we are here to catch botulinum toxin by its horns and become the global leader going forward. The total market in India is something hardly, I think, 18 to 20 million in aesthetics. If you see the global market, it's close to $8 billion US. Countries like Turkey, Thailand, Poland. I'm only mentioning those countries which I say we can compare with our sort of a mindset, population, or in terms of economic strata. I'm not comparing ourselves with US and Europe right now, and not even with Korea and China. Even those countries have reached a potential of 1 million vials. So thing as GUFIC, what we need to invest is to help you all also, and that is where one of the reasons of GROW program, to educate not only, I mean, sorry, nothing to educate you, we cannot do anything, you will actually educate us, but we need you to create more injectors, we need you to create more, I would say, applicable to support a $1.4 billion population. We need many clones of yours. At the same time, we need to create this positive awareness in customers also, which I know that at your level we cannot do it. But if we all of us come together where GUFIC takes care of the financial resources, we need to create the market of botulinum toxin or Stanox, whatever we call it, that there is a requirement. We have to get the stigma out. We need to show that it is having many applications. Right now also we all say, oh, if it's a US and Europe indication approved, then we do it in India. You all are much bigger experts. You all have indications which you have been using off-label, or you will be getting new indications which you will work on, where I want to sit with you. GUFIC wants to sit with you, come out with publications, get them approved by the DCGI, and get indications from India to outside. Why should we be from outside to in? Let India be the ground of innovation. Let India be the ground of being the first user or the first mover to get it outside. So that's the purpose I have. Today for Stanox, hopefully in the next 6 months to 9 months for the new filler, which is also going to be a game changer, and then for meso products. So that's all from my side. I'll not bore you. That's the next cue for me. Today I'm just an executor or a caretaker. The main hero of today's show is, there are two heroes. One is of course Dr. Balram Singh, and then of course Dr. Andrew. Dr. Balram Singh, apart from being my mentor, guru, or I would say my scientific dad, I have a biological dad and a commercial dad and a scientific dad. So he is a PhD from biophysical chemistry from Texas Tech. Of course, he's earlier, I forgot to mention that, yes, I'm mentioning now. He's an Indian, I was an undergrad from JNU. Then he went and got his PhD from Texas Tech. He's the president and CEO of Prime Bio. Of course, he's also the head of founding director of Botulinum Research Center. Just pleased to say that in the entire world, there's only one botulinum toxin symposium which happens in August every year. It happens in Dartmouth, Massachusetts. Under his guidance, under his thing. Even Dr. Alan Scott, who used to be a part of this program till before 2-3 years before he passed away, was one of the, I would say, forefront experts in that symposium. So be it Allergan, be it Merck, be it Ipsen, all of them come together to his symposium. Even the product of Allergan's Botox has a reference to his paper when the actual thing is there. So today, what I'm trying to say is not that I'm trying to be pompous about it, but here we have the source code, or the mothership, as I always say. We have the main guy who wants to now make the product from India outside, and he's an expert in many things, apart from toxin in Ayurvedic biotechnology, neurotoxin research. He's also working on spinal regeneration also, where people who have completely lost their spine. And again, he'll talk much more about it because the relation of gut and brain, I'll not get into all that. And more than 13 books, 300 research articles, and like I said, more importantly, the man and the face behind Stanox and G.R.O.W. So I welcome you, sir, to this podium, and looking to hear from you. Thank you very much.
Hello. Thank you. Okay. Thank you very much, Pranav. He didn't introduce that I'm from a village. He visited a few months ago near Ayodhya. That has a relevance to this story. I could never think that I would ever go even to Delhi, forget about America. But it did happen. When you work hard and you have the kind of mission that I hear, Pranav, I'm so thankful to you all that I could hear some of his stories that I did not know before. It has implication. If I had known that he was so driven for botulinum product, I would have asked him more money. But now I'm glad that his enthusiasm is very, very important for us. And when he came to visit us, I could see the energy that he had, and I see that energy continuing. And that is very important part for this kind of thing that we can do. So I'm going to talk about a little bit philosophical, and I'm primarily an educator, although this is also an education event that is happening. I really, really appreciate people who directly deal with other people, except for politicians. The doctors are the ones who really, I don't know whether you knew this or not, but doctors are most trusted profession, and nurse. And then high school teachers, not professors, because professors only profess. But it's very important that we understand the value of practitioners, because they are the one who can bring the questions. I think there will be some sessions here for questions. I am somebody who, Dr. Andrew Ress, who will be talking, and I'm always eager to collaborate with doctors, because I want to see what is your problem from your point of view, and then maybe I can find some way to address it, which is really very, such a satisfying thing to do. I did not want to go to America. I didn't tell you this yesterday in Delhi. I did not want to go to America. I am Pakka Indian, and I wanted to remain in India. But circumstances changed and I had to. Then I said I will go to America and then come back with the training and do something in India. When I was coming in 1989, I had an offer from JNU as a professor. My postdoc advisor, with whom I learned botulinum toxin, he said, "You're talking about your children." He said, "Why are you going to India?" I said, "I want my children to grow up in India, Indian culture." He said, "But you cannot even afford public schools in Delhi that you are going." Because that time was समय 700 से 1600 की scale हुआ करती थी. So, but he said, you can do from here many things. And that was a great advice, and I really thought that I will do something for India. I live in America, but I'll live for India. And in 2003, I tried to find some business partners to be able to deal with my technology, but I think it was meant to be later on when Pranav joined. And we were able to work. As you can see today, there is a product, and nobody can be happier than I am. And I want to, and I'm so happy to hear that you want bring the innovation from India to the rest of the world. That is very important. So the future with time is in 2021, February, the product was launched, and I was here at that time. And Pranav actually went to Ayodhya. This is in Lucknow. We have a center in Lucknow, which is partnering with GUFIC as well as with some other schools to develop biotechnology in the various aspects of biotechnology, including Ayurvedic biotechnology, and connection between Ayurveda being a philosophy and biotechnology being a science, connection between science and philosophy. And Pranav saw firsthand my village and the village culture, where he and his wife were welcome in my home. I come from a joint family. The joint family is living for 8 generations. Right now it's 8th generation we are running with that. So a lot of things that is, I wouldn't call exactly burden, but the sanskar is from the family, and I'm so happy to work very hard to do that, because I think the family culture of living for say 8 generations, it doesn't come that easy. There is so much other, like you said, your dad said, you go figure it out yourself. It's that kind of moments come so many times. But as a family, India has a philosophy of Vasudhaiva Kutumbakam. So that is a good training for me, and I'm so glad that I'm able to play some role in your journey. So what I want to say, these days especially for last year or so, since artificial intelligence has become so formal, I mean, there's been always there. I want to see whether botulinum is more intelligent or the artificial intelligence that we have today. Botulinum is a very old bacteria produced by Clostridium botulinum that's about over 3 billion years, and it has been around even still. That means it knows how to live. Clostridium, as many of you might be knowing, is anaerobic bacteria, so it was when there was no oxygen, and it is still there when there is oxygen. So that's the kind of a story I want to tell. And Clostridium botulinum is a rod-shaped bacteria. it has, I wonder, I always try to talk about 5 C's. First is Clostridium. Second is a cluster. So it has a gene. I mean, Pranav was talking about, how much you can tolerate where. There's a really lot of, the toxin is here, but the other ones are associated. 1, 2, 3, 4. This is regulator. So 1, 2, 3, 4, and then this breaks into 2 when whatever you are injecting or applying is really, so you have 1, 2, 3, 4, 5 proteins plus toxin. And this is a created, when this is a cluster gene, that means it's being regulated by this, what is known as BOT-R, regulatory gene component. So all of them are produced at the same time. And they form, interestingly, they form a complete complex. So they're all in a different kind of proportion that they come with each other. And why it is a naturally made? Because it forms crystal. Crystal means perfect. When it is, because they all join with each other perfectly, so they come in the form of crystal. And we have found, I wouldn't go into detail, but we have found that when you have this whole group, there is a much less inflammatory cytotoxins when you are applying that to your patients. And that's just based on biochemistry that we have. I try to use this, an analogy, Indian culture, like I said, I'm from a village. Science is no different, believe it or not. It's just same thing, human beings think about that. I mean, Darwin thought that there is evolution. But he was making observations all around the world. And based on that observation, social observation, many of them were social observations, then he eventually found the theory of evolution. Now, of course, that theory of evolution can be supported by genes, because that time there was no gene technology. So I have Indian culture, which is brother-sister relationship. Normally, brothers are very loving to their sisters. The sisters are very caring to their brothers. And so I had this idea that, toxin is a brother, and then there are 5 sisters, and they cooperate with each other, because this is a food poison. When you have a food poison, means you can eat it, it's a protein, and if you eat the protein and it still survives in your stomach, that means there is somebody protecting it. And so these sisters are very important. Why I say this? Clostridium tetani produces tetanus, and tetanus is not a food poison. If you eat the tetanus, it will be digested. So because the sisters are not there. So again you have the gene cluster, again you have the complete, again you have the crystal, but then with the cooperation, you see this food becoming a food poison. It has implication to stability of your product, and also that, what kind of side effects you could have. But something related but different is that we use this idea. I said tetanus is a cousin. In Indian cultures, the sisters also tie Rakhi to its cousins. So Fufere bhai, Chachere bhai, Mausere bhai, sabko Rakhi ban jati hai. That means it protects. And actually we did create, based on that we created a vaccine, oral vaccine for tetanus. I mean, all of us get oral the tetanus vaccine, but we got patent on it, and we are trying to find a way to market it. And we have also used the same idea to create this is tetanus vaccine. We have used the same idea to create a vaccine against TB, for example. How is TB? We also have a COVID vaccine. We also have a dengue vaccine. I think Pranav talked a little bit about it.
The interesting part of using the toxin beyond aesthetics or beyond neurology is you can have a 12-year-old who can be prone to dengue, where we are going to come with oral dengue vaccine, where right now, the Sanofi or the Takeda of the world are giving an injection. But imagine your kid or your family's kid can be protected in the next 6 years, 7 years, when GUFIC comes up with the oral dengue vaccine, which can be taken and ingested, thanks to this technology of, again, botulinum toxin.
I'm trying to prove that this is very intelligent protein. Very intelligent protein. So when you are using it for your patients, be aware of it, very intelligent. You can learn, and you can also utilize it for so many purposes. And so this whole, I said that 3 billion year old Clostridium botulinum, at least 3 billion. And it has these different kind of structures and components that play some role. So we actually, I think that was a very good point, Pranav, you mentioned. So this is tetanus. We make it. Then you know, again, Indian culture is that if you have your brother's friends, you also tie Rakhi to them. Unlike in America, you become girlfriend and boyfriend. Here, the sister-brother relationship is very important. So we have created dengue vaccine because of that, COVID vaccine because of that, and many things that could come in the future. So I want to, there is a interagency botulism research coordination committee that has DOD, this has a White House, this is FDA, USDA, CDC. They have been meeting for 60 years. Last year was the 60th year. And I was invited to give a talk, and I gave this talk of deep science of the continuing evolution of Clostridium botulinum for this production and toxin action. So this is a very simple slide where you can get orally ingested, and then toxin gets, it's very smart. It gets out, it not only protects itself, it also gets out of the intestine and gets into the blood, and then finally finds the nerve cells where it enters, and then only active part, which is known as light chain, I think Pranav mentioned about the light chain and heavy chain. And then it's very interesting. There are several types of botulinum toxin, and they all work together. It's like almost like an army. They all work to stop this neurotransmitter release, going through different processes. Interesting thing is that this bacteria is present. How does it know all this? How does it hit our brain, neurons, being a bacteria outside? No, it's not actually outside. It is present in our colon. And interesting thing is that in our colon, 99.9% are anaerobic bacteria. And Clostridium botulinum, well, I shouldn't say botulinum, Clostridium, Clostridium is on the top. So we are speculating, is my interest in research, as to Clostridium botulinum also is part of our body in the colon. And then we will find out when it produces toxin. Because we know, when I started research, you know, Pranav was talking about how important it is, the whole process. Although I knew everything about it, when I moved to University of Wisconsin, from University of Wisconsin to University of Massachusetts, it took me 9 months to get the toxin first time, even though I knew everything. I had the culture and everything, because the place and the process also matters. So you have to adjust to that, and that's a very important thing we were talking about. So this is present inside our body, and it learns. Our speculation is in the process of evolution, it has learned so much about our brain, our neurons. So based on that, there are many other Clostridium species, as you can see here. So many of them, they are all Clostridium. Many of them are there in our body. And some of them also produce toxin, believe it or not. But the point I'm trying to make is Clostridium species is a very important, it predominates in the large intestine and has so many properties it has learned over a period of time, long period of time, so that it can hit our brain. It has learned about the neurons. So the idea of using, I mean, our brain is so complicated, our peripheral nervous system so complicated, but it has figured out everything. If we figure this out, we can also figure out how to use it in many, many circumstances. So can Clostridium botulinum grow in gut without producing toxin? That's one of the scientific questions I'm interested. It produces so many postbiotics, which is good for our health. There are several, I want to just mention, and I want to maybe exhort you. Please keep making observations as practitioners, because many major discoveries, scientific discoveries and medical discoveries have happened because doctors make observations. And then they look for answers. And I think sometimes there is a collaboration with scientists, sometimes they figure it out themselves. So one thing that is very important in this process to know, I know you cannot read all this, it's just to impress you, that there are connections when you have these bacteria and other things, they have back feedbacks So you have several connections from brain to the gut, but then also they have these bacteria that produce things and that they send the information, including acetate, talking about citric acid. Those things are not just simply to produce the, make the pH, but also they have some roles to play. So, yeah.
So the application why we are again saying that there might be in the future where we might come to you do some trials with the help of BCGI, that just doing an injection on the forehead might lead to certain treatments of maybe depression or maybe anxiety. I'm just giving example. I mean, don't catch me now. So there are indications which are going to be expanded beyond the aesthetic one, and maybe there might be a discovery. Like we were talking to a doctor yesterday, that she said that when I use it on the forehead of some patients, I have seen neurological effects and changes happening in the brain also, and that is an actual approved indication. So this is a way where we say the market can be expanded, which can be, of course, be more moolah for you down the line, but at least I'm saying that it has to start with science. So this is just this is where the gut-brain connection makes sense, and I mean, we are trying to emphasize on that. And the future things, when he talks about more indications which are going to be relevant for you, that is where this is a very important slide. Yeah, sorry to interrupt.
Very, very important. Thank you, Pranav. It's what I was saying, the gut microbiome and brain axis uses 80% of neurotransmitters in our body. So only 10% what we think we know. The other rest of it, the bacteria knows what to do. So it's very smart, and I think I'm very fascinated with this. Every day I think about how to understand how this bacteria works. And I think this was discovered by a doctor, by the way. Botulinum toxin was discovered by a doctor who was taking care of patients, very poor kind of family in Germany. I've been to his house. I mean, he's no more, but they have a museum in his name, Justinus Kerner. And but because he was very caring doctor, that's how he discovered. Because people were buying the kind of rotten sausage, cheap ones, and then when they were dying, then he figured it out. He did actually all the experiments, believe it or not. It's a fascinating story. But the point is that this kind of information can come only when you pay attention. So our 20% we think we are great, 80% is managed by this bacteria. So it's very important. I was invited to talk about this injectable summit there was in Spain, and there they are talking about, you know, all the fillers and everything. It's a huge meeting. It was a huge conference, and they had invited me to talk about this because I have this fascination of imagining, all the time imagining and doing some experiments after that, using experiences with many times doctors, like you will see Dr. Andrew Ress. He's a plastic surgeon, but we had so many things common. Many times we will call and talk to each other for hours. So the bacteria, like I was saying, is somewhere here. Bacteria is here 3 billion years, and we come here about 1 million years. So this has gone through all this. It can deal with everybody here because it has survived. And there is of course, there is a molecular basis. I'm very fascinated with that also, because being a chemist, I like to think how the atom, which, believe it or not, the word comes from Atma. The atom word comes from Atma. Its definition is same, it cannot be destroyed, although now we know it can be destroyed. Atom can be done in electrons, protons, and nuclear particles. But the point is that atom is the base of us. So is our Atma. If we understand that, we can learn so much about our Atma by knowing chemistry. So here are the different types of Clostridium. Like Pranav was talking that they have here type A. Type A is here. Actually, C and D came earlier in evolution. A came here, tetanus is next to each other. Latest one is type E, he also talked about. Type E has certain features, both its protein features, its bacterial features, and also its functional features. Like it can be very fast and for a short period of time, and that is something that GUFIC is very much interested. GUFIC is really, you know, very fascinating because first, I think I was impressed because he got a biotech degree from Johns Hopkins. But I didn't know that he was really coming from a business group whose father didn't give him money. So he had to go figure it out, which is a fantastic way to do that. But the point is that the kind of features that GUFIC is working with in its Armada, that you, not only that, I mean, you as a physician, you will be very happy with whatever needs you may have, but also this will send a message to the rest of the world, which is very, very important. It's not because because of his efforts and his business, but India has always had huge impact on the rest of the world. I see it when I go outside and I see how people are very impressed. Not necessarily that with the biotech industry, but otherwise. So I think biotech industry should go along with it. I was saying, Clostridium is at the bottom of the evolution tree that we talk, because we consider ourselves to be on the top. But it has used its intelligence to figure out how to control us. And now we have figured out how to use it to make people look better. So I think that's what you're trying to do. The basic mechanism on this, like I said earlier, there are some molecular things that is going on, but this diagram shows it the best. There are several proteins, they bind with each other, and they fuse with the plasma membrane and release the neurotransmitter. And then when the toxin breaks those proteins, these proteins, it doesn't happen. This causes paralysis. That's as simple as that. I mean, there are so many interesting things about that. And
yeah, okay. So just to add, why the last slide is a little bit more relevant, because that is also one question which you all have. Why does a toxin take 2 days to activate? Or why does a toxin take 1 day? Or why does it last only for 3 months, 4 months, or 6 months? Or why there's a different response in different patients? Why the pure toxin is something and why the complex makes sense? So all this is based on selecting the right toxin and what is the mechanism of action. Now there are very different mechanisms of actions which are dependent on different form of the toxin which you select of different manufacturers. That's why I would say maybe in the Q&A round we can maybe expand more on this slide, Jyoti, and we can mostly focus that how is, not only forget our brand, how are different brands, every brand has its advantage and disadvantage. I will not say, I will not denigrate any brand, every brand is amazing. We need to understand what is right for your patient, and that is where coming to the mechanism of action, which will give you a much better insight where to use brand A, where to use brand B, depending on the condition. Like some people say, my patient is using the toxin for 3 years, 5 years, 6 years. Either why is the effect going down, or why I require less dose now to have the same effect. It's very interesting when you understand which is the toxin you're using and how does it work in terms of the mechanism of action. So sorry, I'm interrupting.
No.
I'm just wherein I feel it has more clinical relevance, I'll just jump in. Yeah.
I as a educator, the only thing I love is interruptions, because that gives me better idea. Honestly, the question, हमारे यहाँ का उपनिषद जो की प्रथा थी, it was only question answer. So when somebody asks either question or interrupts, it triggers something for me to think about. So I love it, honestly. So what I was going to say is the system, the toxin, the bacteria is all anything which is very flexible is normally, you all know this, our body being flexible, our skin being flexible, then you can stretch it. So is this protein, this is one of our major contribution to the field that happened in 2001. We found that this protein is very flexible, and part of the reason it survives that long is because it's very flexible. It can squeeze itself into the membrane, and then other people, the proteases are looking to digest it. I mean, it's a nerves. Nerves are always very well protected. So there are proteases which will try to find anything that comes from outside. But since it is very flexible, it can hide, and that's how it survives. This is the whole toxin. The same thing we found actually a little bit later with another student, which then we found that even the active enzyme part is also very flexible. And it's called molten globule in technical terms. The molten globule is a well-known phenomenon in protein structure. What I wanted to do is not only that they are flexible, but they vibrate at femtoseconds. These proteins vibrate. So we are really working on that level when we will someday, maybe in my next life also, I'm already determined to work with Pranav. It doesn't matter how many lives we go together, because he already called me a scientific dad, so I can't leave him. So, but in next life means, maybe 10 years, 15 years later, when we have all the technology, it will be very, he also introduced, I have some Ayurveda experience. I'm not a Ayurveda doctor, but I'm really fascinated with the science. So in Ayurveda, they really treat you for the morning differently than in the evening. So when you have your patients who are coming or going for some events or whatever they need, and they come in the morning, you will treat them differently because the physiology is working differently, and this the managing of the.
Can I also add one?
Yeah, yeah.
So if you see the last two slides are very interesting. If you see in this slide, at 37 degrees Celsius, the toxin A is the most stable. And if you see toxin E is stable at almost 45 degrees Celsius. So imagine the master cell line and the concentrate which we use is we have to keep it at minus 70 and minus 20. The product which we send to you, as what has been educated to and told you, is 2 to 8 degree product. But imagine that same product which is at 2 to 8, you're diluting it, putting into a patient's body, where the patient's body's temperature might be anything between 35 to 40 depending on his health. Still it is active for 4 months to 5 months. Why? Because the toxin's relevance and the toxin's maximum stability of exertion of clinical relevance is at 37 degrees. So it's so beautiful. Even though it survives at 2 to 8 or minus 70, but it works the best at 37 degrees Celsius. That's why it's made for humans. Because and it never knew 3 billion years ago that humans will come 1 million years ago where they will be most active at 37 degrees Celsius.
Yeah, exactly. Ajit really knew. Thank you. Thank you. Yes, sure. And this is another way of, classifying whatever I was talking about, C and D being on the origin, and the type E being the latest. Type E being active at 45 to 50 degrees, which means in difficult condition of our body temperature also. Even if you have a fever, you can still get this to work. And now that gives me an idea. Maybe there is a way to get the fever down by having the toxin. So there are really so many aspects of this. Biodiversity, there are several serotypes. Each serotype has subtypes. It's like 4 or 5 of them, each one of them. So it's like total of about 50, 60 subtypes, and we are working on it to find out which one is the most suitable and for what. It is unprecedented longevity, survival of a protein. The proteins normally last only for a few hours in somebody else's body. But this lasts for months. It has to be something very special, and we can learn so much from it. I already said that it targets the process, not the particular molecule. The whole process, as if like somebody's had a military strategy. And finally, I told this is very flexible, and that's part of the reason why it can be so long-lasting. It can survive. We have also, so we have done some more work using neuroblastoma cells and all that to find out what else it does. We have found that actually these other proteins, which otherwise are not active, supposedly not active, but this is a evolved bacteria. It would not produce anything for nothing. So we need to learn. We are learning more and more about it. And we have found one of the thing that we have already found, that these associated proteins, in addition to saving it from digestion, also help it to go inside the cell. So you will find the kind of associated protein that we are using that is helpful to you in how you are using
Just to add, there might be different layers of the skin. Just to give you an example, depending on selecting the right extract of the toxin, we can come up with delivery systems of the right peptide or the right tattva, let's put it, which you want to get to the different layers of the skin, and sometimes in therapeutic use, even to at a cellular level we can take. So that is one more research which is happening in the background at Prime Bio also.
Yeah, yes, both DNA expression as well as physiological cytokine response. One of the evidence of what Pranav just said is that when you have the toxin with the complex, the cytokine response is very different than when you have either pure toxin or the associated proteins, which means they are working together biologically. That's very important, those kind of things. This is just to show you, they have been crystallized and other things. And this was first time isolated in 1948 as a crystal. So crystal is a part of the process that we do at GUFIC. I just want to take 1 minute to make a point that the crystal and solution structures are very important. There's another toxin that I worked when I was in University of Wisconsin, and we found the crystals are a little bit different. Because in physiology, with the amount of protein that you're using, there is no way it's acting as a crystal. So as solution structure is very different than crystal structure. To understand your use, we have to be very sensitive to the solution that you are using, how you are diluting, how you are injecting. For me, crystal is very good because we can see the structure of it. But there is a difference in those two. And there are like what kind of buffer you are using, lidocaine you are using, all kind of things will also make difference. And your observations will be helpful to us and GUFIC for sure.
So they will be in the Q&A, we will be covering more rounds on how antibody response is there to toxins. If we are going to work on a new product, which is a patent of his, of lidocaine application in your toxin. Lidocaine might not only be for pain. Like a lot of people feel if I add lidocaine, it will only help me for pain. But you will be surprised to know lidocaine works on the toxin itself to make it more potent. And if you can just try it, use any of the brands, and of course, I'm using your patent and just saying it with your permission. Just add a lidocaine and we'll tell you the exact percentage and the amount of ml. You will see the response will be amplified. And we'll show you papers for that. This is just some new secret, and we have many more. We'll keep on express, and we will be sharing with you in the times to come. Sorry,
yeah,
sir,
thank you. No, I just say maybe another 30 seconds or so. What I want to say that all this interesting evolution, smartness, intelligence that we are talking about, we as a human beings learning from this, that other people have also used, and sometimes we can use to counter that also. So for example, this year's physics and chemistry Nobel Prize went to artificial intelligence. They were using that they can predict the folding of a protein based on the crystal structure database. But I just told you that it's useless, because as far as you are concerned, first of all, the protein at 31 degrees, they never crystallize. Number two is the kind of concentration you are using. Number three, the kind of solvents you are using. That's going to be more relevant. So I don't have a solution, but I have questioned the Nobel Prize winning this year, how careful we should be. Not use that, because there are a lot of issues with that. And that idea comes from this. Botulinum toxin, endopeptidase is the activity, and we have found that botulinum toxin is so flexible, so vibrant, that we need to understand that part. And in that, from that point of view, I say that from artificial intelligence to botulinum intelligence. Thank you very much. I think these are not as important. We already talked about all those. Okay. These are you're talking about a symposium. It happens every year. Thank you.
So we'll need you to introduce Dr. Andrew.
Oh, Andrew, are you there? Can we hear him?
I'm here.
He said
On the other side of the world.
Yep. Yeah. All right. Very good. Andrew Ress is a good friend. I learned so much from him, and I like to share his enthusiasm as much as he does. I always try to learn. I mean, he is a very well-educated individual who is very fascinated. I mean, his degree is an MD degree from University of Miami School of Medicine and all that. But he's also organizes events. Latest thing we have some patterns together also. The latest thing he was very passionate about COVID, and we really worked to try to help people for the COVID also. And interestingly, GUFIC was our mediator in that. They really tried to help us. So Andrew is a plastic surgeon, and he is here. And you don't want to really introduce plastic surgeon with so many words, because they know how they look. Already, Andrew, take it over.
Okay. Thank you, Bahram. We can go ahead with the first slide. Thank you so much for having me at this conference. And, the botulinum neurotoxin has been a major interest of mine, and how I got connected with Bahram was in 2002. We've known each other over 20 some odd years. I was newer in my practice, had Botox that was going bad, even though we refrigerated. It just wasn't that busy. So the question came up is, how do we buffer this? How do we reconstitute it differently so it can preserve the shelf life? And that led me to discover Bahram, and we did some research together, and that led to a patent, which we'll talk about. So my background is, I majored in microbiology and history at Cornell, Miami for med school, Mayo Clinic in general surgery, then spent a year doing research in plastic surgery. You think, what would that be about? But we, in reconstructive work, did a lot of peripheral nerve regeneration research, ischemia reperfusion, injury investigations, things like that. And then I'm certified by the American Board of Plastic Surgery and on the faculty at Nova Southeastern University. Our research resulted in a patent being granted in 2010. And what we found was that um, we could reconstitute Botox or and any botulinum product with a different diluent rather than saline. Okay, and in fact, next slide. Next slide, please. There we go. And in fact, one of the things that we discovered is that sodium chloride actually causes early dissociation of the heavy chain and light chain, with the light chain, of course, being the active part of the toxin, and causes loss of activity early. And in fact, when the manufacturing process that Allergan uses, um, it has has a gram of salt. It's doped with salt. What are the other things we found out? That Bont is more stable at lower pH and also more stable after reconstitution if you keep it in higher concentrations. And in my practice, I dilute it with the least amount of saline to keep it as concentrated as possible, and then further dilute it out right before administering to patients. Now, um, one of the things that lidocaine, how would that act? Well, lidocaine, the pK of lidocaine is like 5.7. So it's acidic. So it may be the lidocaine, it may also just be the pH, but that's something to look at. Clinically, I'm not sure I would do that because lidocaine burns a little bit, and patients are at least here, very sensitive to any kind of discomfort, especially when they, with saline, it's fine. Next slide. So at the beginning of the pandemic, it was looked like from the in vitro data in lung cell cultures that hydroxychloroquine when inhibited uptake, and of course this is just in vitro, but Bauram and I looked at using hydroxychloroquine as for pre-exposure prophylaxis, and we were able to get a prospective randomized trial organized and through the FDA and the pre-IND process. At some point after that, the emergency use application had been revoked, so we were able to go ahead with the clinical study. But I think it's the importance is to ask the questions and being able to do the experiments, whether it be in the lab or clinically, to get answers. Next slide. Okay, well, botulinum neurotoxin is one of the most useful drugs ever discovered. It works on all muscle fiber types, both striated, smooth, peripheral nerves. We talked about the central effect. Is it from retrograde transport or persistence of the toxin? Who knows? Could the toxin be bound with other agents and have more central effect? It's certainly possible. It's a magic bullet designed to hit nerves. Next. So we have now the ability to customize the results of using botulinum neurotoxin for whatever you're hoping to achieve. And this is culturally specific. And you think about how you want to change the patient's appearance, what they want. And now we have the tools at our disposal, whether it be minimally invasive, non-invasive, and ultimately surgery. Next. So I've used neurotoxins in my practice since 2000. Primarily, I use at this point Dysport until we have Stunux available in the United States. But I've used all these products, and as you know from your practice, they each have their own characteristics, both in how they handle, dilute, and work with patients. So what are the major products? Well, we've had an explosion of new products out there. I think that Stunux is going to be a hit once it hits the American market, which hopefully won't be too far out. Um, if you look, especially on the where it says in the column it says stabilizers and excipients, you notice that, and it's a type which should be 0.9 gram, that Botox in the first thing is um, has a huge amount of sodium chloride, and and then Dysport, which has a good amount of human serum albumin, has a tendency to spread more. Botox spreads the one that's the pure toxin, meaning no, just the light chain alone, which is Xeomin, that spreads the least. So each of these products have their own spreading characteristics based on the osmolarity and protein load, and immuno inducive characteristics. And so with Stunux, we've learned that the purification techniques from prior, remember the first batch of Botox was created in the late 1980s. And so they didn't have the purification methods, nor did they know. So now we know the least amount of serum albumin, protein load, all those things, the better the clinical effect. And in fact, uh, so we talked about spreadability. Dysport spreads the most, but again, you just uh, modify your technique. I find with the spreadability, I can inject Dysport further apart and have fewer injection sites, whereas with Xeomin, you have to inject more closer together and have more injections. Next. And we talked about the units. Um, the Botox is the standard. Well, first to go back, there are no international units. Each product, which means it's standardized between products. It's, in fact, um, each product has its own unit formations based on that LD50 technique. And so um, with Stunux is done in has the same as Botox, but it really has to do at the end of the day with the amount of toxin that you're injecting. And we're talking about picograms of active toxin. Picograms. Next. So where do we get into issues? Well, you want to make sure that you don't put in so much toxin that you elicit an immune response. Otherwise, it will cause the toxin not to work in the future. Now, it may be with different subtypes. Let's say you have a patient that says they have antibodies. There is a test for it, but you can try a different type A product to double check. And unfortunately, if they do have antibodies to the type A product, then you're going to have to go to the uh, type B, which will get around it doesn't work as much, as well, and as long. Okay. Now, in general, the other thing is the frequency of injections. That creates a higher risk of an immune response. And the immune response can be against the non-functional NAT proteins, uh, or functional where it impairs the light chain. And that's where the ones were. So it doesn't matter if you have an antibody response to the non-functional proteins. It's if you have light chain binding. And typically in our cosmetic situations, you're not going to see it because we don't put that much toxin in, but you should be aware of it. Next. I had a dermatologist, this is about 10 years ago, I had a colleague of mine do my neurotoxin, and this is what actual ptosis looks like. You talk about local side effects. A lot of people say, get injected and their brows get heavy. It's like, I have eyelid prop. Nope. I get to take out my driver's license and show them this picture. This is what real eyelid ptosis looks like. And if you look at the anatomy of the orbital rim, on the superiorly, there's the orbital septum and membrane that is a mechanical barrier to spread of the toxin from the frontalis muscle. It's really injecting the procerus is where you're getting the spread. You don't have that mechanical barrier to block the spread. If you're injecting platysma muscle, you can end up with dysphagia. And if you're injecting the masseter or the crow's feet, you can end up with smile deformation either by affecting the other muscles. So the zygomaticus major, if you're getting too deep by the orbicularis oculi, or by getting the risorius muscle in the masseter area. Next. Okay, so where have I used it as a help me out tool? After face, and I don't know how many plastic surgeons we have in the audience, but neuropraxia after facelift is reasonably common, and it usually resolves on its own within 6 weeks, but they'll have asymmetry of smile. So it's very easy to administer some Bont in order to match the asymmetry on both sides, and a patient can go on their lives while the nerve regenerates. Can correct the smile asymmetry, brow asymmetry if there's weakness of the frontal branch. Rarely occurs you have a salivary leak. Injecting it in the parotid or submandibular gland area will dry it up, and you don't need to use anti-salalogs. Next. So treatment of eyelid ptosis, apriclonidine drops, and you can even use a little double-sided clear tape. Dysphagia is important to make sure that the patient isn't at risk for aspiration. And in that most extreme case, you may need to keep them NPO and put a feeding tube in. But again, unusual in the cosmetic arena, more common when we're treating cervical dystonia, injecting a large amount of toxin. Bruising is probably the most common complication. And next, I've come up, especially around the eyes. Next slide. And next slide. And I've come up with a technique that I'm in the process of publishing, where I keep 1 cc syringes with lidocaine with epinephrine with 30 gauge needles, and as soon as you start seeing that little bit of purplish come up, I put a wheel of local anesthetic on either side of the vessel to mechanically block it, and then epinephrine kicks in and causes it to vasoconstrict, and it really has helped limit the extent of bruising in this area, which can be quite annoying. Next. Wrinkle scoring. I think everyone is familiar with the MER scale. Sometimes it's good to be able to have we like to classify everything. So this is just another way of classifying the severity of wrinkles. And it's easily available online. Next. And this is just an example of the typical Likert type scale, and it's applied pretty much over the face. This molecule can be used anywhere in the body and has been. Some of the newer uses, we talked about salivary gland leaks, hyperhidrosis, axillary, palms, soles of feet, facial sweating, facial oil. Every day there's a new indication, and more will be found. This is Raynaud's. I've used it in patients with bad Raynaud's and vasculitis. Can make a difference. Next. Well, what are the newer uses that we're doing here in the United States? Well, platysma, horizontal necklines, patients that have a dynamic nasal tip when they tip moves up and down, nose moves when they're talking, can inject the depressor septi nasalis muscle, which is right underneath the columella, gummy smile correction, lip flip. So without using fillers, being able to rotate the mucosal portion upwards in order to have it look the appearance of more lip fullness without having to put in filler. Now they got to be careful to only get the superficial fibers right at the vermilion border, otherwise you're risking having a asymmetric smile or weakness. Next. And here's some examples of using it in the platysma. And doing all these non-surgical things are helpful until the point where they're ready to go ahead and have a facelift. Next. And then you go back to maintenance with the toxin. Here's where you treat for gummy smile. And next is example of the effect of it, and then as it wears off, it returns. Next. And other weirder uses. Yes, scrotox, injected in the scrotum, and I'm not going to show you before and afters, I think, for everyone's relief. One of the unusual, the slide's not showing well, facts I was able to find is it's that the amount of bot that you need to inject in patients in a sunnier, warm climate is higher, statistically significantly so, than areas that are colder, darker. So it's an interesting observation. I'm not sure it makes that much of a difference what you do, but it's something to be aware of. Next. Now, occasionally you'll see a patient, and I had this happen a couple of years ago with a patient I gave Dysport to, that several weeks later started developing all these unusual neurologic symptoms. And anatomically, they didn't make any sense. She would end up having to be hospitalized. They wanted to blame the neurotoxin. But in fact, from an anatomic standpoint, could have only been one of two things, myasthenia gravis or multiple sclerosis, and it turned out to be myasthenia. Okay, all these autoimmune diseases can either be unroofed and expressed by an injection of neurotoxin, doesn't matter which type, or, and that is why it's also, or flare after getting any, and that's why it's botulinum neurotoxin is contraindicated in patients with these autoimmune neurologic diseases. And it's just, again, another something in the back of your mind, so you know to identify it and get the patient to proper health care. Next. I want to thank you and look forward to your questions.
And I think with so much of learning of Hello. Yeah.
Yes.
So
Okay,
yeah,
good.
yeah. Thank you so much, Dr. S. I think it was very insightful, and with so much of understanding of botulinum toxin in depth and understanding the science, it's very important to now understand very quickly the Indian patient's experiences, our panelists' experiences into understanding how clinical outcomes are very similar or differ compared to our global exposures of patients' outcome. So we have a very interesting panel discussion, which is going to be led and moderated by Dr. Madhuri Agarwal. So just a quick introduction. Actually, Dr. Madhuri doesn't need an introduction. So Dr. Madhuri is a founder and medical director for Yavna Aesthetics Clinic. medical head for Yavana Academy of Aesthetics Medicine. And with lot of accolades under her umbrella, I would soon want to invite Dr. Madhuri to invite other panelists. Thank you.
Thank you so much, Dr. Jyoti, for that kind introduction. No, I think we all are hungry. That's what me and Dr. Satish were discussing. Though maybe this, I am sorry to disappoint, but maybe this may be a little shorter than we expect. Okay, so I'm glad to, do I go to for the introduction to the next slide? Okay. So I would like to next invite the man himself, Dr. Balram Singh. So it's a pleasure to hear you, and I think a lot of philosophical things, I think that gut and brain connection that we all as dermatologists talk, you kind of summed it up for me. I think that's my takeaway today. And it's an honor to have you on the panel. I'll try to do justice. Then next up is Dr. Satish Bhatia. He's a dear friend. I've known him. He's done extensive work in injectables, not only toxin, and so much to learn from him today. We also have Dr. Rixon Pereira as our panelist. Okay. Dr. Rixon Pereira, again, a good friend. He's doing amazing work, and we also see a lot of good technologies coming from you, Rixon. So I'm going to learn some things today. And last but not the least, Dr. Devraj Som, again, known him for years. So a pleasure to have you, Dr. Som. He has multiple publications to his credit, and again, I see a lot of good work coming from you. So thank you so much, and I'm sure is Dr. Andrew joining us for the panel online? Okay. Okay. So.
I'm here.
Okay. The panelists here need no further introduction. That's why I've kept it very short and brief, and we'll go to the main panel. Can I have my slides here, please? Can I get it? Yeah. As Karan Johar says, I'm getting the coffee with me, with my glasses, budhape ka glasses on. Though with Stenox, we can never get old. So I guess everybody in the room has been using Stenox for some time. You better say yes. We have still dinner to get to. I have been using it since the launch, and I trust, initially when the brand came to me, I was very wary. How is it, as Pranav said, that is it Indian brand? How will Indian doctors take it, ये तो घर का ही है, made in India है, थोड़ी अच्छा होगा, बाहर का ही अच्छा होता है. But to my surprise, I was proven wrong, and I saw amazing results comparable to international brands. Till we load the presentation, would you all want to, Dr. Satish, Dr. Rixon and Dr. Som, can we have the mics for our panelists? Would you want to add your, initial expressions or reactions to this brand when you all got it? Can you get one more?
So I have been using Stanox for I think close to 3.5 years now, and I think I'm almost exclusively using it
Nice.
since the last I think 2.5 years. And my first patient, of course, I had received a sample vial and always try everything once. And my first patient was a patient of masseters. So I said I was not entirely sure of the upper face. So I started, I said, let me just do masseters because, the per unit cost is lesser. So I said, let the patient benefit from it. And I use the standard dose that I use for Botox, which is 30 units per side. And after 3 to 4 weeks, her masseters actually got really, they really shrank to the point where she got a jowl. And that's when I realized this thing is really powerful, and you cannot you should sort of regulate the use. You cannot, if you know that it's powerful, then you have to not be so nonchalant about its use. You sort of regulate, understand the toxin. So I started using upper face, and I started with lower doses, and then sort of found my own dosing protocol for Stanox. And in a lot of patients who had, because we were doing Botox before for years, and I found that the results weren't lasting as long. And by the end of the third month, people were like, it's gone. We need to, and that's when I started doing Stanox in those, and I found really long-lasting results, almost for 4 to 5 months easily. And that's how I slowly shifted to Stanox.
Stanox
Yeah.
exclusivity. Good. Dr. Satish, any?
Yeah. So I started using it about 5 to 6 years ago when it was launched itself. And the first thing I wanted to be always careful. I wouldn't go on to masseters. I wanted to go on the upper face itself. But interestingly, I would say is I found the cost effectiveness as compared to the other X brand. And B is the onset of action. Within 2, maximum 3 days, patients started saying that they started seeing the results. So this would be the key point for using Stanox.
So I think sasta, tikau, effective, मजबूत भी. Dr. Singh, आपने सारी checklist कर दी हमारी पूरी. मुझे commission देना. So sorry, how do I make this go ahead? Sorry, we are technically challenged today. So Dr. Som you are remaining to add your bit about your initial reaction when you used Stanox.
I started using botulinum toxin from my fellowship days in the US, which was about 2 decades ago, and was almost a complete Botox user. Then we started using Xeomin because with the incobotulinum toxin, the advantage was that the temperature sensitivity went away. So as a result of which you could transport it across centers. And then Stanox came in. Now, if I look at my fridge, there is Botox, there's Xeomin, there's also Stanox. But I think in majority of our patients, we use the Stanox. Botox as botulinum toxin went off patent. And the moment it went off patent and it became a generic, at that moment in time, what started making sense was how much effect does it create and at how cost effective is it? Because ultimately, it's our job to make our businesses more proficient in terms of profitability. And that's where Stanox has done a really good job. So I would say, like Rixon, my practice is now about in fact, at the aesthetic clinics, at all our centers, 70% plus is Stanox.
Nice. I hope Pranav and Vijay is hearing this. So yes, I hope we see more studies like this. There's this study which was shared, and I was very impressed. I think this was shared quite some time back with us, wherein they compared it to an international brand, and they treated quite a good number, 50 patients with moderate to severe crow's feet, and they found that almost comparable results with this toxin. Stanox showed comparable results with onset of actions within 2 days and lasting up to 4 to 6 months. 4 months is what they saw. So it is an endorsement of what the brand talks about and what we as injectors, like I have experienced it, Dr. Rixon, Dr. Satish, Dr. Som has spoken about, and many more out here. So let's get on to the panel discussion. We all know India is the major population, the next superpower, I believe, and I think I'm glad people, innovators and entrepreneurs like Pranav and are coming up and adding to that vision and making us globally out there. So I'm not going to talk already. Vijay has mentioned the figures. As doctors, we go with the figures. So why is Indian face different? I think this is Bella Hadid. She was labeled as the most beautiful aesthetic face some time back. What are the differences you see in your patients, Dr. Rixon, Dr. Satish, Dr. Som? You all can pitch in. How is it the same as the international standards, or are we a little different?
I think we have, we have more square faces. We have less oval faces. The mandible, but it also varies, like from the north to the south, and from the west to the east. So we have more wider faces. The mandible, the gonial, the bigonial distance is also prominent in a lot of patients. Then, other than that, I don't think, of course the skin tone, the thickness of the skin that's higher, it's more seborrheic, so that you have more oil gland activity, so you see more pores. And because of the good collagen degradation is a little slower, so we don't see the onset of lines really early. I mean, in younger patients, we rarely do the upper face indications. Those we really start doing, because I've seen some of my Caucasian patients, they do it in their late 20s. You know, second, in the later half of the 20s, and with Indians, at least for upper face lines, you know, we usually start in the 40s. You know, that's early 40s is when people start doing it. So I think, yeah.
You want to add anything, Dr. Som?
I wanted to.
Is it working? The mic?
I wanted to actually talk on 2 points. First, the paper which has been published in the Journal of Cutaneous and Aesthetic Surgery. It's a great product, but the product probably deserved a better paper. It's a low impact factor open source journal. 1.05 is the impact factor. More importantly, when it was being used, there was no double blinding. So the people who were injecting knew that they were injecting Stanox on the right side or the other one on the left side, but they knew which one it was. Should have been double blinded. If we had done all of that, it could probably have been published in a much higher impact factor journal, especially when you read that the article itself was sort of funded by the company itself, and it's open to all sorts of interpretations. The article could have been better, in my opinion. Now, the second thing that I'd like to say is we actually, in 2019, at the PRS, and Dr. Rees is American Board Certified Plastic Surgeon, but that's the official journal of the American Society of Plastic Surgeons, PRS, and we published How Indian Faces Age. So this was a photographic study of 300 Indian patients taken from the west, the east, the north, and the south of India, which actually took their photographs across 6 decades. And this longitudinal photographic study actually showed us how Indians age. And what we found was very interesting. What we found was that even though Indians are very varied from the north to the south and the east to the west, one thing is they don't develop the wrinkles, like Rickson said, in the back of the neck or in the top of the forehead as much as the Caucasian skins do. Because we have melanin, we are mostly type 3, type 4, type 5 skin, and that there is also thicker skin which prevents the wrinkles from developing in that area. So one of the major reasons, in my opinion, why the aesthetic industry for botulinum toxins has not grown at the way Allergan thought it would 2 decades ago is because India is not Botox country. You don't really develop wrinkles in the upper part of the face. India is more filler, threads, Althera, devices, facelift sort of country, because it's really the mid face, the lower face, and the neck where you're losing a lot of volume. There's a lot of descent which is occurring, and you're beginning to lose definition. Also, the anthropometric measurements of our skull are such that our our mandible is actually much smaller than the Caucasian mandible. So the support system is not there. In some sense, the bed is really small, and the bed sheet is becoming larger and larger because we are putting on weight. So in some sense, the real advantage for Indian companies developing botulinum toxin, or any of us developing a botulinum toxin practice, will come in when we start using botulinum toxin in indications where the usage of botulinum toxin is much higher. For instance, we had a Miss India contestant come in recently, and she wanted her trapezius made smaller. And it's an off-label indication, but putting in 80 units into the trapezius, which is 160 units, is something that we would never be able to do, because no matter how much you did on the upper face, you wouldn't cross a 40 to 50 units. So those are some of the elements of India which make the expansion of the botulinum toxin market a little bit more challenging in terms of the physiology and anthropometry.
Agreed. So yeah, so we don't fit in the Indian bracket. We have our own beauties. So, Dr. Satish, you want to add what is the typical aging? I think they've already touched based on it. Do you want to add anything to it?
I'm just going a little beyond India.
Okay.
Okay. So I get a lot of Gulf patients, and what my per observation I've seen is they have their upper face, they have a much thicker muscle contraction and hypertrophy.
Yeah.
So, and Stanox has worked well for them.
So I think we see varied population, and it is applicable to, I'm sure here we have experts who are also sitting and they have their own exposures. But you're right, in metro cities like us, we see less of upper face. But we do, in our clinics, do share a bit of masseter toxins, jawline now people are coming asking for it. We do a lot of Mentalis, DAO, Gummy Smile. So, other indications than the conventional is what we do. And yes, hyperhidrosis, and now it is going to the Barbie talks, as they call it, and a lot of other indications we are going to talk of.
Maybe
So
I can ask question to Andy because he's just waiting there.
yes.
Andy, you live in Florida. There is a lot of sun there. Do you really see a difference between people who are living in the New England area and then Florida in your practice?
Not particularly. I think it's if you look hard enough, there probably is, but
Did you ever have Indian patients?
I do. Not a lot, but I have we're a very multicultural area in the United States, so we have you name it. I think you just have to listen to your patient, and and everyone's different.
Okay, thank you.
So I'm coming to Dr. Andrew now. So we spoke a little bit about Indian beauty. When we're looking at at Europe and US and Indian, is there a difference in perception since you're dealing with both cultures out there? Do you see there's a difference of perception in the population there of toxin? Like a patient perception, is there a difference?
Well, it's extremely well accepted here in the United States. We have people are talking about pre-tox, so getting it younger and younger as a prevention strategy. And it's not just about getting it, they want to try the different brands, it's who do you go to for it, what are you using, and can I get the result that I want?
Okay. Any specific treatment goals when patients come to you, Dr. Dixon, again the Indian panel here, Dr. Satish, Dr. Som? Is it different, the perception? Is it easier to convert nowadays because of the awareness? And is there a treatment goal you as a physician look at when these patients come to you?
Actually, there is still a sort of a negative emotion attached to doing injectables, and patients usually ask, what can I do for these? And then the moment you suggest an injectable, they're like, oh, am I too, is it, can I wait a few more years, or I'll do it when I'm 50, and those kind of, and a lot of this has to do with what has been shown in the media. They don't know the difference because you see botched filler jobs.
Yes.
And people assume it's Botox.
Yes.
So they constantly need this one's Botox. So they assume that it's botulinum toxin, which makes their face swell and look big.
Yes.
Whereas it's mainly botched filler job. So there is still, and then you have to still explain to them that there's a difference, because people don't know the fundamental difference between toxin and fillers, and what can lead to this. And of course, then there are a lot of, in the cities, urban areas, a lot of people are already well read, and they come and they say, I just want to do this. Is this an option for me? I've read about it, because social media is, of course, increasing awareness.
Yes, sorry.
So I'm going to go on the opposite side.
Yeah.
Is probably during COVID or just post-COVID, everyone, the kids, between the 20s and 30s, they started coming.
So
we want preventative Botox.
Preemptive, that's what he was talking about. So you shifted the spectrum, you came to the younger generation.
The younger generation.
So I think a little bit I would want to ask Pranav, we as doctors do a bit, to talk about our patients, about the awareness, and as Dr. Dixon rightly said, patients come presuming that this is filler and not toxin. So as a company, what do we look at from you all to increase this patient awareness, partnering with doctors besides the product? How can we grow this? Is there any move there or any plans there?
So I'll just talk loudly. No, so I think basically what I think Dr. Dixon said also is the Indian requirement of indications, because I think, okay, thanks, sorry, I'm very technology challenged. So the Indian requirement has to be much more, I would say, specified. I think there are other indications. And what I would say as a company, apart from using our money in terms of raising awareness, which of course we will do, and that's what we are committed to because we make it, we also want to work with doctors like you all, create our white papers, like you rightly said. Till now, maybe we did not do in the right paper or the right patient study or the sample set, but we want to work with each and every one of you all, create the use in different indications which are more India-centric.
True.
And actually make a difference. So that's why more and more doctors can easily use it. That's always a hanging sword, off-label, hai, ye, hai, wo. Also, one more thing, just to add, I think for the forecasting in the future, and since you mentioned Karan Johar, so if you see we work on semaglutide, and we're also going to come up with Mounjaro's generic down the line. So that is one big thing is going to help all of us also going forward.
And
But
he's
of course,
already
I think
helping
doctors
us.
are much more. But yes, I think on a serious note, once again, we as a company need to not only create more above the line, below the line awareness, actually bring the all as front force to talk about this openly, remove the stigma, what Dr. Pereira rightly said, that stigma should get away.
Yes.
And more importantly, create that awareness that, I would say there are multiple options done. Hyperhidrosis is one thing which is very less done in India, which I
True.
feel it's a big
Market here in India.
And again, like you said, come up with more papers by which we know that there is more relevance to that with more awareness. So that's
So
right.
you know
sorry.
whom to connect. Yeah, so, sorry, this is gone. So patients' expectation, as Dr. Rickson rightly said, this is what they see in the news and they say, oh my God, I don't want results like these. But, what is happening there? So, Dr. Som, coming to you, what are your expectations, from a good toxin brand in terms of speed, efficacy, and longevity? We have Dr. Singh here, we have Pranav and Dr. Andrew there. So we're going to ask them our wish list.
The ideal botulinum toxin would be one which acted through Bluetooth. You didn't have to inject.
You have now set Pranav on a journey. He's going to do it. I can see it.
Bluetooth or Wi-Fi, so you don't need needles entering the face.
Next year, he's going to be standing and talking. That Som led me to this.
Most people would like to look better, but without needles entering their face.
True.
They don't mind needles entering their scalp, but they don't want it entering their face.
True.
So if there's a way to do it Bluetooth, that would be interesting. If it lasted forever and put facial surgeons like us out of business, that would be even better. But there are toxins like Daxify which are coming up, which are lasting much longer. But there's nothing called a perfect toxin.
Dr. Andrew, is there a truth to this? Because we have not used Daxify. Do you see it lasting longer than 6 months? Have you used Daxify?
No, I haven't used it yet.
Okay.
But the fewer, of course, the patients are going to want fewer injections.
Yeah.
But on the other hand, it keeps them coming in the door, and if it's, and you're maintaining that relationship. So I think 3 months, people are here used to having the longer acting. Helps, but I don't think it's a critical thing. People are just so used to coming in every 3 months.
Dr. Singh, please, we would
Yeah,
want you to add.
I was just going to say, because Dirk Dressler, and you probably know Dirk, right? Dirk Dressler from Germany.
Mm-hmm.
So he has done some research on it, and I checked it out. So Daxify went for FDA approval, asking for all this, and FDA refused, because they are using double the dose normally people use.
Ah.
So they think, and Dirk Dressler has done some research on it, and he presented at IBRCC last year. And he thinks that the longer lasting is just because you're giving double dose.
Oh, okay.
So
Sure.
I think this is being sold, from my understanding, my connections telling me that it's not selling as much as it used to in the beginning, because people are realizing. Why? Because they're afraid, and some of you talked about it, that maybe there will be an antibody production more likely because you're using so much of material.
So,
I think that's the immediate concern I have when you said it's double the protein load essentially.
so that's the secret now we know. Dr. Singh, this is a whole list of questions for you. You have already spoken about them, but why Sternox? Why should doctors go for Sternox? Is it different toxin? Is it very pure? Is it very stable? What are your thoughts as an inventor on this? How do we as doctors take this?
Yeah, I really only know what I do, and we make it the best technology that's currently available. That's one. The formulation is slightly different, but more importantly for Indian doctors, I think my sense of Ayurvedic principle that it is produced here locally, and it is in the environment that is people are used to, we're talking about somebody who gets more sun, it has difference than those who have a fairer skin. The same thing is now, how would that be possible? I mean, Dr. Som probably will be having objection to that. Because he is somebody who has done so much of this work. But how does it do it? I think I mentioned that because it is how it is. Right now it's working. The question is how. Of course, our technology of producing is very clean compared to other people. I have seen this, how the industrial companies work. They have a protocol and they follow, and they're required to follow exactly the protocol. There are a lot of changes, including the gene that they have, when they have a culture, several terms going on. We have done and we have published actually some work. How if you do it, at one time we did 210 iterations. For 210 days we did this culture and a culture and a culture. And we found that 13 mutations naturally appeared in that. So most of these companies don't pay attention to that kind of thing. We analyze everything because we are more interested in the science. So this is probably the only, I say Xeomin is, because there was a scientist, Freebert, Dr. Freebert, who was very heavily involved. I think this is probably the best combination of research and a company with energetic CEO who wants to make difference. And I think this event, I'm so impressed with Dr. Jyoti Jha to have this in India, so that you can interact and you can give me some ideas, asking me this kind of questions. The way this might be happening, what I was saying is that the protein structure, even it changes. The people think that, oh, well, maybe we maintain the temperature, but you don't maintain the elevation. There is a difference in the pressure, and that this protein can get affected by that because it's so flexible. So all those things are out there to find out and learn more about it. But the fact that it is working better than Botox, the process is pretty much the same. We just use a little bit different label or time of dialysis or something like that. Doctor, because
maybe we can add specifically about citric acid and also.
Oh yeah, the good point.
So I was going to drag you in the panel. Yeah. You are an unofficial panelist, anyways. I was going to ask you that if you Indian doctors have to talk about this.
Yeah.
So I think what he's talking about citric acid. These are citric acid is an organic molecule, and like I said, this molecule has developed over 3 billion years. So there was a question, Satish ji was asking about lidocaine. So lidocaine, we use like millimolar concentration of lidocaine, and we think it was a solvent until somebody recently who wants to, I told this to Pranav, who wants to license this technology, said that, tell me how exactly it happened. What about like citric acid? We found, even though you have a lot of citric acid, but there are certain areas, and we have not done citric acid, but we have done lidocaine. Lidocaine goes and binds to near the active site, even though there is plenty of other things. And we have, I'm disclosing it, although we haven't patented yet, but the activity doubles because of your lidocaine. Same thing is citric acid. What happens in these movements of the different domains of the protein, that either they break or they denature. But if you have certain areas covered by something like citric acid, it keeps it in the same place.
Yeah,
and
absolutely.
active.
So I think just to be very specific, when what Dr. Singh perfectly, Daxify, I don't want to name any of the brands, but just by loading the toxin is not the solution, because you want a patient to come to you for the next 10, 20, 30, 40 years, depending when they start the process. So when I mentioned during my talk also, we need to come up with a formulation which is having the right dose, not leading to the side effects, at the same time having the potency you require.
True.
So the combination of right now, of course, citric acid, down the line, of course, with lidocaine, and then we want to come up with that 6 to 8 months period for you, only if you want. Because I think he mentioned a very important point, Dr. Russ, that you want to see your patient every 4 months.
Yes.
If they come after 6 to 8 months, there are a lot of other baggages which get lost in the entire process, which you don't want to do that. So you want that 3 to 4 touch point. But however, if you still feel that you want someone for the 6 to 8 months, it should not by doubling up the toxin.
True.
It should be the right toxin with the right excipient, which helps in binding more, and which is safer for you long term. So that's what I would say, that as Stanox or the future versions of Stanox which we come up with, we'll always ensure the safety and the toxicity is taken care
So
of.
we have audience questions. Can we take it, Dr. Jyoti? Dr. Gayatri wants to.
I want to ask, what about zinc? They say zinc also prolongs the effect of.
Dr. Singh, would you want
Yeah.
to?
Well, the zinc is part of the active site, and sometimes you lose it. So if your preparation was made in such a way that you lost some of the zinc, it depends again what kind of other materials you are using. For example, EDTA is one of the, tetraacetate is one of those. If it did remove it, then you adding zinc will be helpful. So it depends on
Basically the
storage condition.
So actually zinc, I'll tell you my sense, I may be wrong, and just to say, so if you see zinc orally always helps, depend on the viability of the formulation, it helps to keep the healing or the overall processes in common. Like we always say zinc is excellent in healing. But in terms of binding, we have never come across papers which actually say that zinc reaches to that concentration in the cellular level by which directly helps. So, but now when we talk about lidocaine is actually something which is going along with your product and clearly working, and of course with the science and the papers and all, we'll share with you with actual diagrams and 3D programs, where it's actually helping binding the protein to your receptors without actually modifying them. So you don't want a muscle tonicity to happen. You don't want that the free frozen phase to happen down the line. Or I hope I'm using the right words. What is there? But very frankly, zinc, we don't know that whether we have any paper that orally it makes much of it.
Yeah.
Locally also, I don't know. Maybe
I misunderstood. I thought zinc with the formulation. So yeah, zinc does have metabolic effects because of its ionization and the size. There is somebody who recently wrote, because I also work with bhasmas, and zinc bhasma is different than zinc. So there is ionic, zinc is very important to maintain certain types of structure in different kind of.
So I think we can take offline Dr. Singh's opinion on
I
this.
will be happy to.
because-
I will be happy to.
So this is a paper which we had published on Indian aging skin, which says are we aging early? We already talked about patients' profile concerns. Dosing, I think this is very important. I think Dr. Som, Dr. Rickson and Dr. Satish, can you each answer like what is the dosing pattern generally? Has it changed over the years? And we talked about repetition of sessions. Are the patients open to coming regularly? Do you have that percentage? So any one of you can take this up.
I think the dosing pattern remains the same, 2.5. And I personally haven't changed the dose of dilution.
Okay.
And the traditional one, whatever you have for the upper and the mid face.
So
will
Dr. Som, I
remain the
wanted your two opinion. There are now these lot of speculation upper face only 3-point works and the new techniques. What is your take, since you also do a lot of publication and research?
We use Botox, botulinum toxin pretty much the same way as we used to earlier. Obviously, botulinum toxins are such that they work biologically, so they are not interchangeable. If I would use Xeomin, then I would use it at a slightly higher point above the brow because it tends to diffuse more. Similarly, for the Ipsen toxins, that's the same, it diffuses more. But short of that, it's almost the same. It's remained the same for a long period of time, and nothing much has really changed, at least in my practice.
So I think the conventional take is, that we are doing what we were doing initially, maybe a little bit here and there, but I think there are publications, we should take it with a pinch of salt. Yeah,
I have a question. So all you doctors, did you actually feel as the patient comes to you in year 3, 5 or so on, does the dose required, do you need to titrate it anyway?
So I have blepharospasm and hemifacial spasm patients who've been literally following up for the last 15 years, coming every 3 monthly and getting approximately 60 units of Botox. It's remained almost the same. In fact, if you went ahead and looked at them, the fact that you've been using the Botox so regularly has actually led to atrophy of those muscles, like the corrugators, etc. So they actually have reduced the spasm. We are writing a paper now, how using botulinum toxin in blepharospasm and hemifacial spasm leads to long-term biological effects by which the impact of the Botox starts to last longer. In fact, very interestingly, 2018, MERS came into us. We run a CRO as well. So MERS came into us because they called themselves the pure toxin. And they said, we want you to talk about how we are the pure toxin. There's just this 150 kilodalton molecule. There's no complexing proteins. You should talk about how it works in people who have antibodies. So I said, I haven't seen botulinum toxin with antibodies. There have been about 10,000 plus patients we've done with botulinum toxin. There are 500 plus who are repeating every 3 months, and we still haven't seen antibodies. So how can I speak about something which I haven't seen? You tend to think that because there's albumin, there's complexing proteins, there would be antibodies, but in clinical practice, the impact of those has been literally very poor. Whenever people tell us that antibodies are resulting in no effect, it's usually poor storage of the toxin and poor injection technique.
Yeah,
I think
I agree with
I fully,
him.
because even
Yes.
Dr. Dirk Dressler, who came from Germany, actually echoed the same thing, that there's no antibodies, which he has faced almost in 35 years of practice.
Yes.
And more importantly, like you rightly said, a couple of doctors we met internationally also, and of course in India, they also mentioned the use of the amount of toxin eventually after some years is actually reduced. I mean, you need to reduce because of the perfect point. I think perfect point.
So
right?
that's what we experience with our masseter patients. When they do it very religiously, then you can actually see the spacing out of results. Dr. Dixon, you wanted to add something?
Dosing patterns remain the same, but what I've seen with Stanox is the results last longer. So dose to dose. If
So what do you mean by
combine with
last
conventional,
longer?
Usually I see with other toxins, it usually comes back by 4 months, and this probably gives you an additional month of longevity.
At the same dosage?
Dose to dose, yeah.
Okay,
Dose to
fine.
Same dose.
Dr. Satish, any difference in male versus female? Does your dose vary? Does your injection points vary? I know it's a very traditional question, but we have beginners here, so I thought let me bring this up.
Yeah, but okay, in general, between male and females, obviously the males, if you're doing the upper phase, the dose would be slightly higher. But again, it's different from person to person.
But Dr. Som, again, don't we say that we want to preserve some lines in males, and do you follow?
Sorry, I wanted to add also. And also another thing in females, we are adding a few, say, 2 units each besides the corrugators in the depressor septi also, to give them
that
nice
brow,
flare.
yeah.
The brow lift.
Whenever we inject anywhere around the eyes, we will impact the shape of the brows. Now the only question is whether we are impacting them positively, in line with their gender alignment, or we are impacting it adversely. In general, in females, we would want the lateral arch of the brow to go up. And if the lateral arch of the brow to go up, if we want that to go up, I don't inject Botox beyond this particular point above the brow, because I want the frontalis to actually pull this up in this manner. But if you did the same thing in men, then you would have everyone looking like Amir Khan in PK, and
That's
that
a
wouldn't
Spock
really
point.
look that great. And so as a result of which, it's important to go right till the tail of the brow to ensure that the brow stays flat, because all our male icons, whether that's been a Keanu Reeves or that's been a Sean Connery, have had deep, intense eyes. No one looks good with a flared brow.
Absolutely,
so short
I
of
agree.
that, everything else is
We
fine.
like our men to be intense. We don't want them feminine, not like Korea. So just quickly, I'm showing pictures. You know, when patients come to us, I talk to my patients about expressions, not about lines, which is what Indians like. So we talked about stressed look. We talked about tired look. We talk also about angry look. Nobody likes to look angry even if they're not. We talk, you know, with whether it's male or female. So I think that helps a lot in our practice. I don't know whether you all do that, but we talk more about emotions because we Indians are emotional. And I don't think we talk about, you know, you got lines. They will say, अच्छा बाद में आता हूँ, अभी तो मैं 30 का ही हूँ, बाद में हो जाएगा, you know, that way. So, do Indians age early? I just want to quickly touch base on that preventive. I know you have a study you spoke about. This is a study which we published about, you know, Indian skin, and we realized, yes, we do have patients coming earlier on. Like I have male patients, actually more than females, who come in 26, 27 years saying that I've got these deep forehead lines, doc, and I want to get rid of it. It's genetically there. So, any experience with preventive toxin, you know, when people come to you, do you do it? Do you ignore it?
I don't encourage it.
Why is that?
I think somewhere we have to not follow any hype, and there's nothing called as prevention. I mean, if you see a line, treat it. If it's not there, why assume that it's going to come and try and prevent it? So I'm not a big fan of prejuvenation or preemptive toxin. But once the lines begin to appear and a patient is concerned, then we have something. And see, with antibodies and all, we never know. You start early on in life, and maybe your body it may stop working after a few years. So the longer, the longer you delay it, the better.
Yeah. Dr. Somi, you have that smile on your face.
I think everyone in this room is living evidence of the fact that preventive toxin works. So some people may be inhaling it, the rest are injecting
Pranav,
it.
are you looking at Pranav? Yeah, I also thought
so
such.
it's preventive.
He's literally inhaling it now, sorry.
Preventive toxin works. Now let's talk of our brows. everything that every day mother gravity is pulling us down. So there's descent. Descent is the major part of aging. But with that, there's also muscular action which is pulling it down. If you could stop that working at age 20 and impact the brows positively, at age 50, you may not need a brow lift. So preventive toxin is about having your vitamins in time to prevent you from having the major problem in the later. It's like a vaccine.
Yeah.
And that's what this room demonstrates, that it works.
Dr. Singh, I wanted to ask this out of curiosity. That as Dr. Rickson said, that when you don't have lines, why should you toxin? Is it really true, or should we start injecting the moment you feel you have hit your 25, 26 as people come to us? He's the scientist.
I know it won't work on me because I'm immunized when I started working on it. But I think there may be, actually, especially what I'm hearing, that there is no antibody response. If you want to do preventive, you may be, actually there was a article that I read a few years ago. There is some actress in London who had started getting her daughter, who was 16-year-old, injected, so that she will never have to face this. Maybe there will be a day, maybe what you were saying, that you never have to face wrinkles.
I think more of the static thing also.
Yeah.
Some people believe that if it becomes static, then it's not
Yeah.
working. So just take care of it before, right? I think that's what was one of the
Yeah.
Dr. Andrew, do you do that? Do you see patients in mid-20s and though they don't have lines, do you suggest injecting them, or how do you do your preventive toxin?
I usually see patients, the youngest are late teens, early
Wow.
20s. And it's really driven by the social media.
I think we have to all look at Pranav to do this. We want that kind of buzz in India.
awareness we have to.
True. So I agree with Rickson, Dr. Rickson, that when you have lines, or the beginning of the lines, maybe you can look at that. But I personally believe when there are no lines, what are you going to chase there? Maybe use your devices then, okay? So these are some of the results. We do a lot of masseters, as I said, and not much of both male and female. Very gratifying results. They keep coming back. Gummy smile is again an underrated, and we do many more indications like mentalis and AO and necklines, and nowadays Nefertiti again. This again is something which you're doing more often. Lip flip, as Dr. Andrew mentioned. With just little few units, you can actually give an effect of a lip filler, though it doesn't last long, but patients don't mind because it's very small amount, cheaper than a filler. And again, it's used in body, the trap talks, as they call it. Again, I would want to ask Dr. Satish, do you see this? I think you do quite a bit of this, right?
Yes, I do quite a bit, but going back to your previous slide on lip flip, we've done a study on close to 35 patients, and we had given them the option whether you want to do a filler or a toxin. Basically, it was more of a counseling, and then when it came to finances, the lip flip turned out to be much cheaper with much more natural results.
Absolutely.
So,
Yeah, that's...
and trap talks, yes, I do 1 or 2 in a month.
See, we Indians are G.R.O.W. We find things where it is not there, Dr. Som. Up ऊपर नहीं तो बाकी सब जगह चलेगी. So yeah, this was very, amazing, shocking. I kept asking Dr. Jyoti and Ravi, ये कैसे हुआ? How did they grow hair? I'm sure it is not toxic, it's something else, and they showed me the results. So have you all seen this kind of experience in your practice?
I haven't tried it.
No,
for
no.
androgenetic alopecia.
Now, Dr. Jyoti, you have to pitch
Too
in and tell
expensive
us.
to try.
This is a result which is only with Stenox injection, where these patients have been coming every 3 to 4 months' time, and it's very, very gratifying to see this result. And we are very proud that one of the doctors is going to be publishing this. She has done 50 patients
Wow.
with result like this, and you can show more results actually, which is amazing. See, like this kind of result.
So I
So-
can see Dr. Umesh, my dear friend, who is a hair transplant hardcore. What are your thoughts on this? Do you think your hair transplant patients will stop coming to you after this?
Actually, hair transplant has its own place. Obviously, hair follicles will be intact, and because of what blood supply we are improving, those will those hair will improve, but definitely hair transplant will have its own place.
But
Always.
do you think you would be tempted to try this in your practice?
Absolutely, this wonderful result. Wherever we are seeing miniaturization, we can definitely go ahead with this.
Yeah.
Yeah.
Yeah. I'm definitely going to go back and try this, because I think what I understood from you, it's equivalent to a PRP, dosage, and maybe Ravi can share with the doctors here what are the protocols, because it would be really, yeah.
We might be bothering you to collect them and actually go for a publication by which you can go to DCGI to actually make it an official indication, provided it's in the purview and in the right thing.
So this is off-label at this moment,
Yes.
right?
Now, but that's what we want to, like I said, we want to work on many indications which are off-label right now and make India the ground to make it official going
How
forward.
are we diluting this? We are diluting it as 10 ml in 100 units, is it?
Regular constitution is what they told us.
And how many spots do you inject? And
So
do
the
you-
entire
do you do a
scalp, I
grid?
think
Do
you
do
do
an
around.
Apache?
Yeah, so it's entire scalp, like you are basically covering the entire area which is affected, and approximately around 50 to 60 units per time, and in each point you are putting around 2 to 3 units. So maximum we can go up to 5 units, but 2 to 3 units has given this kind of results.
And it is repeated once in 3 months.
3 to 6 months is the patients have come for follow-up, and this is a result.
So
It's
we all are going to do a study for you guys.
Yes,
Pranav.
yes, I think that's the next thing to call
Have you
for.
done a monotherapy? Is it just
This is, so we have mixed patients, but 50% of the patients are only on Stanox. So we have a data of 50 patients almost.
So I have one question. How did whoever was doing it
Yeah.
think that this would work for hair? There is one paper. Never read it.
You
There is
have,
one paper.
Ravi, please get up. He's the man you have to chase for all the data.
He's the man on whom Stanox won't work on the head.
That's why I highlighted.
So
That's
yeah.
why I highlighted it. You better get it done, Ravi. Otherwise, people are going to question
So
you.
no, the whole
Ravi,
idea.
if it works on you, that's his 3rd billion.
Yeah, so there's a protocol, okay, which we'll be sharing with the doctors so that they
Sure.
all can try and give their feedbacks to us.
Yeah.
It's very, very interesting.
In the meanwhile, you're also trying to figure out the actual mechanism of action, because tomorrow when we go to DCGI, we need to have some pharmacology act also. So we already have some breakthrough, but some other.
No, actually, yesterday Dr. Balram had mentioned about the anti-inflammatory action, and because of which how the entire scalp inflammation can be calmed down, along with obviously the blood circulation and stuff. So yeah.
Yeah.
Dilation of the vessel by
Yes.
proliferation.
Yes.
Yeah.
I think I had seen maybe 10 years ago,
You want me to?
Allergan had done this experiment and presented in one of the conferences. I mean, there could be multiple mechanisms. One of the mechanism is that to lose the hair, it has to squeeze the muscle as a part of it losing the hair. So this relaxes and it cannot squeeze the muscle, and that's why you do not lose the hair. But there may be other things. Actually, we have some formulation that we are working on in this process, especially with the topical formulation that we
yeah.
have.
So just last few more slides to go. I know Dr.
Yeah, we
Satish
can-
has an early flight.
Yeah.
So,
we can just have this last question.
Yes.
actually.
So toxin fillers, devices. I get this asked by a lot of juniors. I ask by patients. How do you do this? Do you do it on the same day? Do you space it out? You all can all add your bit.
Not on the same area maybe, but
Okay.
if it's on different areas of the face, then we can do it.
What about you, Dr. So?
We've done everything on the same day.
And the same area.
On the same area as well. Sometimes there are
Wonderful.
patients who are coming in from overseas. They're coming
Yes.
in for 4 hours.
Yes.
And we had no option. We've even done laser on top of the Botox, which has been done, like CO2 laser, etc.
And didn't affect your
It's been
results.
exactly the same.
Yeah.
Nothing's really
I
bothered
believe,
them.
I think there was a study published wherein the HIFU and toxin and fillers on the same area was done, and it kind of was patent, but I still am little guarded on it. I still have my reservation. Dr. Bhatia, you do a lot of overseas. What are your
Yeah,
protocols?
so if at all I have a hitch is between doing fillers and threads together at the same time.
I agree. I've seen necrosis happening there, but otherwise, yeah. Yeah, so to conclude, we have our own unique faces and we customize our protocols. We see the differences. And as Dr. Singh said, hum Indians hai, hamari bhartiyata hum rakhte hain. Dr. Singh, any last comments before we wind up?
I think the one aspect that didn't come up is the medical tourism or aesthetic tourism. If you have your product that is here, a lot of people from outside would love to come. So you want to not only present it for Indians, but also I think Pranav and we are working. There are various ways to work on that. Is one of them is the FDA approved product, FDA approved facility, but it's still using the plastic surgeon or dermatologist from India. So that people will be coming here for a variety of other regions, but also we'd like to do this. And that will be a good thing for India's influence throughout the world, because you're changing their face. Can you imagine that? You're changing their face literally and metaphorically. That will be a wonderful thing to have. I asked the dermatologist and plastic surgeon in India. I know Andrew Ress is also looking for this product. Maybe we will import him here as well.
Yeah, we would love to have him here. We'll make him Indian.
But this will be really a great thing to do. If we got the product that is, as you are all saying, that is really so good, I think we need to find a way to use it in various conditions and also promote it for people to come here.
Thank you so much, and I invite you all to YAM ADAC 2025, where Sternox hands-on workshops are there. So I think there's one question out there. We can take that.
I think we can use Botox for preventive scarring.
Yes.
Yeah, that is.
Unfortunately, we're short on time. We had a whole list of indications. But yes, we can talk it offline. Yes.
I want to know how to reverse,
ptosis.
Not only ptosis, but supposing, I've heard about saline injections reversing or reducing the effect, supposing.
Complications of toxin, can it be reversed by saline is what doctor is asking.
I've seen some.
Normally people use antibodies at this point. My lab has a couple of antidotes which work against botulinum. There are a couple other people who have those kind of only not approved drugs yet, but antibodies are approved. So antibodies, if you really have, most of the time people think that it's long lasting because it's several months. So to neutralize, just use antibodies if you get the complications.
You wanted to add, Dr. Som?
There's nothing you can do. It's chemical denervation. Until the acetylcholine receptors come back, there's nothing that you can really end up doing. So
Dr. Singh is working on the antidote is what we said. So hopefully we'll get something soon.
We screened 51,000 compounds and we identified 4 of them. We published that paper, and because of that paper I get a lot of calls on people who have been affected because they say, when is this going to be a drug? I think when I convince Pranav to spend the money on it, there will be a drug someday. We have the compound. It's just to make it a drug, it costs a lot of money, and unless there's not that much the business in it. So that's why, unless maybe government gives money.
Unless if you're a good injector, you won't have that.
Yeah, that may be the best thing to do.
Yeah, thank you so much to wonderful panelists, and thank you Pranav, Ravi and Vijay and Jyoti for giving us this opportunity to interact.
We want to thank you for, moderating such wonderful session. And we just need another 5 minutes from all of you. So we want to take this opportunity to first of all felicitate Dr. Balram Singh, sir, because I think without you, this entire scientific symposium wouldn't be a possibility. So yeah, please, Mr. Pranav, if you can just felicitate, sir.
Sure. This is my, I said, infinity plus one shawl on your shoulder.
Yeah. Where is Dr. Please, you also please be here. Yeah. I would now request Dr. Balram Singh, sir, to please felicitate our panelist and our moderator, please. This is Dr. Madhuri, please.
You can come in the center.
I know we all are hungry. Can you please clap? And we also want a group picture, so please wait for a few minutes. Yeah? Dr. Sir, please. Dr. Satish. Dr. Debraj, thank you for insightful discussion. Last but not the least, Dr. Rickson Pereira. Huh?
He's such a curious researcher.
Yeah, he is. because he added a lot.
I found that when you clap, it affects your skin. It helps, and also it creates innate immunity.
We have to clap for the immunity. Yeah, and Dr. Rickson Pereira. So clapping increases your immunity. So now you all can clap. Yeah. I would request Mr. Pranav and Vijay and please come on the stage for the group photograph. And I would request all of you, thank you for your time, at this hour. And if we can have a group photograph, please. All of you all, please come on the stage. Stage or dais, yeah. Yeah, first, all the panelists, please come. One group picture. So, please come. All of you, please join. Team, you all can also join. Avi, Harshara, please come now for the picture. Okay.